Hey, good morning, everyone. A very warm welcome to you on the second day of this wonderful symposium. Thanks to all the speakers for making yesterday a great day. So today I'm going to chair this session along with Mazia Ibrahim. I'm not going to introduce myself, but I'm just going to quickly introduce Mazia. Dr. Mazia has a PhD in computational biology from Anna University. She started her career at IITM as a postdoc researcher with me, and now she's a project scientist in the incentive project at IBSE. Her research interests are in bioinformatics and computational systems biology approaches to analyze microbiomes and multi-omic data to understand metabolic behavior in microbial and human systems. And with that, I'm very happy to introduce our keynote speaker for today. I think Venkat's already been introduced once, but let me just add a few things. I think if all the youngsters in the audience, if you haven't had a chance to look at his biography, please go pick it up. There are copies in my lab if you want to reach out. And I think it's very inspiring to read his story. So Venkat's research spans from the deepest reaches of the ocean to the unique microgravity environment aboard the ISS. His pursuit of understanding microbial life in Earth's most extreme environments as well as extraterrestrial settings highlights his exceptional contributions to astrobiology and planetary protection. He's been very closely associated with several Indian institutes, with IISc, IIT Madras, and so on, and he is very eager on giving back. So I encourage all of you to take up this opportunity, grab him, get mentored by him. He's always eager. Thanks, Venkat. With that, please.
Thank you, Karthik, for the hearing. Thank you, Karthik, for the introduction. And if you switch off everything, people can sleep. That's not a problem. Very good morning to all of you. Thanks for coming so early. In India, it is so early, but not in our place where we are. We started the program 8 a.m., but here is roughly 9:30 or 10 o'clock, because I think the biggest thing is traffic. You guys are all first-hand exposed to the traffic. For 1 kilometer, you have to wait for 30 minutes or 1 hour. Lucky here, Madras, but if you come to, it is not Madras anymore, it's Chennai. If you come to other part of the India, like Bangalore and Delhi, it's worse than this. So you are blessed to have a better traffic here. Okay, so today, I'm going to take you to the other places. So let me try to take you to space. But there are so many other things that we did for the past 25 plus years in JPL about microbial monitoring, but I am going to talk about one of the things that is different from other things. So this always I put as a first slide. Some of you have seen this before, but just to remind you why I'm doing this. So this is Sojourner 1996. We sent to the Mars to look for some geological stuff, because after Allen Hill meteorite, which NASA prematurely said we found life in other planet, Mars. But some of the researchers who did, including my mentor Ken Nielsen, was putting all the calculations together and said even one genome cannot be in that particular size. Even the smallest genome cannot be put into that size. So they were telling we need samples back to confirm these kind of things rather than the meteorite, because the structure and physical thing only cannot prove the life. So you need to have both to become the biology. So for that you need the sample. So this was just 1 kilogram weight, less than a kilogram weight, okay? And when you are going to other planet to look for evidence of life, you cannot contaminate the other planet, because today's technology may not be good. Limit of detection is very, very high, but 10 years from now it will be different. So what that means is NASA come up with an international policy, which is a gentle human policy, which is called UN Treaty in 1967, said that any planet that you are exploring should be carefully, thoroughly taken care and not contaminating things, because at least 50 years of your lifetime, you will have some different technology to look for, so better not to contaminate. So we come up with a committee on space research that inclusive of all different spacefaring nations, including India, that actually abide with gentle human policy. There is no gentleman anymore because we have gentlewomen as well. So that's why I always telling gentle human policy here. So we adhere to that policy, which is not policing by other country. If you are sending it, it is your responsibility to tell, yeah, I took this much amount of the microbial load to there. So I'll explain you in a bit later what the microbial load that we are coming here. We are looking into spore as a proxy for cleanliness of the spacecraft that is flying and landing in other planet. So any federal agencies take food regulation, drug regulation, medical requirements, and everything is come up with some sort of quantifiable number. So without that, the project manager cannot, clean to the level that is desirable. So in 1975, people come with so much of research to come up with this spore, which is, again, humanly possible measurement that you can do. We didn't have the shotgun metagenomics then. So come up with an idea saying that culturable aerobic spore formers, which are the one that can survive harsh interplanetary conditions, desiccation, radiation, you can think about, low temperature, etc., etc. So they come up with a thing called 300 spores per meter square, not to exceed 10 to the 5 per lander. Lander inclusive of this rover. The rover is the one that's roving, but lander is going to land the Mars. So that inclusive of that massive structure together, I'll show you in a bit later, should not contain. If you open a lander, for example, it will be a one big football field size surface. So one big football size surface should not exceed 10 to the 5 aerobic spore former. We are all extremobiologists here. We know, it is not only spore can survive, so many other things survive, but we have to come up with some sort of number that is humanly possible. Otherwise, you will not fly. Okay, this is the thing. But since 1997, now 2020, this is the same thing. This is Mars Science Lab, Curiosity. But what we have right now is Perseverance. So all those things are not taking only the small, but it is going from here to golf cart size to Mini Cooper size. So this is 1 ton size. 1 ton size with the lander mass is one cricket field or one football field size if you open it into a 2D structure. Okay, that should not exceed 10 to the 6. It's 10 to the 5 in total. Okay, but all the 10 to the 5 spores cannot be in one pocket. It should be evenly cleaned. That's why 300 meters per meter square. Okay, what I'm going to do is today is unlocking extremophiles hidden in NASA spacecraft assembly clean room. So one of the things that we all get bombarded with the National Research Council, Space Studies Board, with the full of academicians and other scientists who are involved in these different agencies are telling, NASA, you are not living in DNA era. You are still doing the good old spore-based assay. Why? Let's do this shotgun metagenomics. Let me come into that in a second. So I'm Venkat. I'm retired from JPL and now adjunct professor at the University of North Dakota. I'm going to show you these what is associated with these spacecraft surfaces, and also take it to the International Space Station, some of the microbes that are actually resistant to the space conditions, and then with the help of some of the space payload, we can expose it outside the space station and then come up with some sort of thing that is even good for our Mother Earth. Okay, not only the microbes are good for space exploration as well, and also you have to give back to that because it is taxpayers' money. Okay, so I always run over. That's why I want to give credit first to all the people that are working. It is not only one person thing. That is, I'm still hungry, doing a lot of different things, and you see some of the familiar faces here. Karthik and his students are here. What is the main thing? So whenever I'm talking that I'm from NASA, they will be asking this question: why microbiology in NASA? It is an engineering institution. What the hell you guys are doing there? Because you are looking life in other planet, which is robotic mission. When you're looking life in other planets, better don't take life with you. That's simple. Okay, there is a contamination control. The second one is, that is what the planetary protection means. That is, if you go to a place where you know life is, then it is better to preserve that particular environment rather than putting something from 10 years, or your grandkids may have a better technology to go there, and then that will be a kind of contaminating the one that already you explored 50 years back. But don't ask me what Elon Musk is going to do, but that will be a different story. And that's what this is, human mission. So NASA is interested to take human there. That means sustainability of human presence in other planet. What do we want? We want oxygen. We want food. We want to live in a better condition, protected from radiation. All those things can be elucidated with the help of the microbes, how those things survive in other planetary condition, then you can exploit for in a human research program. Can you make oxygen with the help of microbes, which people are doing it even on the earth as well. So can you use the microbes to grow the plant with the resources that you have it there, inside to resource utilization? You cannot take tons of earth material, soil up there to make them grow there, right? So one of the biggest thing is, as I already told you, we are in a DNA era, right? Why the hell we are just doing the spores? How can we use the metagenomics to come up with some sort of requirement that NASA can get a clean spacecraft before flying? Okay? Are we missing these culturable microbes in the metagenomics? Because we are all bragging that we can get anything, everything from metagenomics. These are true with the rare microbes. Different people have different perspective about the rare microbes. If you're asking the deep sea microbiologists, they say a lot of dark matter out there that you cannot resolve. That is the rare biosphere. I'm talking about low level of microbes that you can culture in a petri dish. Are you covering in metagenomics? That's the one, okay? Here is a video that how we are doing collecting samples and then using it. It is a 2-3 minute video. Just check it. I'm not going to talk in between.
It's really important that we send a rover that's clean and we make sure that it doesn't contaminate Mars. My name is Mujige Stricker and I protect Mars from Earth bacteria. The next Mars rover is slated to go to Mars, collect samples so that eventually we can bring those samples back to Earth and determine for the very first time, did life exist on Mars? There's nothing that we can build that's sterile. So we take swabs and wipes of the spacecraft as it's being built. It gets put in an oven, it gets put in various chambers and clean rooms so that we can maintain that level of cleanliness. If we do find something on Mars, we have to make sure it's something that actually came from Mars and not something that hitched a ride. This is the place where the magic happens. Oh, it's definitely cool. In this lab, we look specifically at spores. So spores are those hardy microorganisms that can actually survive if it made it on the spacecraft in the journey through space, through the vacuum. It's very humbling to be a part of this big project because there are hundreds of people that have to come together and build a spacecraft. There's no one person that could say, I did this, I made this happen. It's always a we. I owe it all to Carl Sagan and watching the cosmos. I remember being a little kid, going to the public library and renting that VHS. And from that moment, the light bulb turned on. It actually was the start of my passion of science communication. We are citizens of our universe. We have to be good ambassadors when we are exploring other planets, other moons. So it's the right thing to do.
So to take that particular 1 ton weight of the rover, you have an enormous big lander system. Okay, all of them has to be taken care in terms of microbial burden.
It's really
Okay, let me break it down what I say that, So under the current NASA standard process, what you do is take the sample, grow them, and identify them using 16S or other gene marker, and also archive them for various different purposes. So that what we do first is we are looking for spores, I said. So take the sample and heat shock 80 degree 15 minutes to kill all the vegetative cells, because those are the labile microbes, may not survive in space or transportation as well, interplanetary transfer as well. Okay, look for the number of spores that are aerobic, I told you, but there are a lot of anaerobic spores are there, and that we are not considering because it's made up of some requirements, and project managers has to come up with some sort of number. So this is better, but still it is not faster, okay? 2 to 7 days, and then count them, and then pick them representative colonies and further identification. But that's not what happened since 1992, because NASA has, the Space Study Board as well as National Research Council insisted that spore assay only will not give the enough bioburden. So we did non-spore assay, but a culture again, and also we were doing some gene markers like ATP. LAL is another one, is called endotoxin, that is used in medical industry to test the intravenous fluids that we are all administered, which is the LAL assay. And then quantitative PCR, you guys know. So these were also looked into to see whether this can give the cleanliness of the surface of the spacecraft before flying. Okay, because spore assay takes 7 days. So the schedule is so tight, they cannot wait. If they build or assemble one top of another phases, then if the number is exceeding, they have to dismantle and redo it, recleaning it. Okay, so this will be a quick study to tell you how much is there, so that you will have a confidence. But here, dead and alive will also come. That is a big issue. And then we also did cloning and sequencing. This is 1990s, so we did those 16S-based clonings also. And after that, with the Lawrence Berkeley, we did the phylochip analysis as well. And then we got the 454 and Illumina. So this one is NGS, as you all know, and we all do right now the nanopore as well. So long reads, PacBio, everything. PacBio is for the known microbes, but the nanopore and Illumina are the ones that we are doing right now. So this is there, but we came a long way in terms of microbial detection here. But still, the standard assay requirement is based on number of spores. Can we detect them? Okay, so when I say rare microbiome here, don't think, can you get confused with the rare biosphere. That is two different things. It is what I'm trying to say, very low abundance, but still persisting over the period of years. So metagenome sequencing in NASA mission critical cleanroom environments encountered challenges in detecting extremophiles due to their resilience and low abundance. We are cleaning the spacecraft assembly facility. We are cleaning the spacecraft to the limit of detection of per today's metagenome approach. Somebody can say, hey, I have a multiple displacement amplification. I have a whole genome amplification. But that is not necessary. We are not drawing blood out of the rock. We want to follow and see whether we are missing or not of the required target microbes, which is spore here. So those are less than 0.1% of total microbial burden. When you get the reads, first thing what you do is anything more than 1% of the OTU only you take. That means those are the dominant players, That's what we think for that particular ecosystem that is doing something. We forget the one that is less than 1%, which is catalytic for certain environment. Specifically NASA, all these spore formers are almost you cannot detect. The stringent decontamination measures on the nutrient sky, it is we are not building a spacecraft in our garage. It is actually in a clean room, low nutrients. We call them oligotrophic environment. Okay, nutrient scarcity resulted in less metabolically active extremophiles. That's what I was asking with Jennifer yesterday. Can we do SIP technique, stable isotope techniques to look for specifically some spores? But she already answered correctly that even though it is permeable, this will not go in. So a lot of issues are there to measure that. So leading to underrepresentation in shotgun metagenome sequencing data. The robust nature of extremophiles, spores are extremophile. Particularly spores protected their DNA, complicating extraction and potentially compromising the genetic material quality. So additionally, their genetic material are often overshadowed by more. This is the biggest problem. We are getting the microbes that are not relevant for NASA mission. Because human is still going, so human is still shedding. We are walking fermenters, so we are actually shedding a lot of microbes that cannot withstand space conditions. So with the help of Alex Rosado from COST, we did this study. So this one is culturomics. One Phoenix mission that we did, one of the intermediate rover that I showed you, that is the Phoenix mission, 2007. We exposed collected not only spore formers, but also mesophiles, thermophiles, sacrophiles, acidophiles, alkaliphiles, halophiles, anaerobic, 5% hydrogen peroxide, because these are the ones that we are using right now to decontaminate the spacecraft component. And then it is exposed to UV. All these things done, you collect the samples exposed to these conditions. Okay, so that is to some extent. How many conditions I can give? 1,000? It is not humanly possible. Okay, so let's see how it goes. So this Phoenix mission, we got around 215 isolates out of these things, and then did DNA extraction, whole genome sequencing, quality check, all the kind of things. You got it, and then we got 53 strains out of 215 were novel, consists of 26 novel species from one mission only, 26 novel species. You go to your garden soil, I bet you get only 1 to 5% of the total microbes that you are collecting will be novel because of the dominant microbes. Okay. Then this one, new term for you guys. We have not introduced this. When you're going for extracting DNA, you get both dead and live. So as yesterday's speaker, she was using stabilized isotope. She was also doing some genomic thing about origin of Odyssey level to see whether that is live or not. But what we do is we collect the samples and then treat with propadium monoazide that actually block the DNA from amplification. So only the live cells that you have not touched by PMA, and that is allowed, that is available for your further amplification. So that is PMA metagenomics, but during 2007, we all know we don't have a shotgun metagenomics, but here I had some 200, 300 samples of 2016 of the same clean rooms that we were able to get the metagenomics. So we got this metagenomic DNA treated with PMA 164 samples here, raw reads, filtering, mapping with the novel species. We got the novel species genome. We mapped that with this to see whether anything that is from the pool of the metagenomic data is there or not. So assembly is going there as well to look for what are the things that are present in general, what are the things that are dominant. But these novel species strain, which we have this genome, also look into this and map to see how much is actually present in there. So presence and abundance of this novel species in the clean room metagenome, okay? So I'm going to show you Phoenix as well as Mars 2020 rover thing. With the help of, Pratyay and Karthik student, we did this work. So bacterial diversity of NASA clean rooms based on culturomics first. So the one that I showed you before, 10 different conditions, or that come up with this kind of 26 novel species, comprised of half of them are spore formers, half of them are not spore former, but withstanding that condition that I showed you, okay? So let's look into that map. This is the whole genome, tree based on those, whole genome tree of those 26 novel species here. It is nothing novel, nothing fancy here. The biggest thing is 25% of the bacterial strains, which I told you 215 isolates we have, worked on, were novel. That consists of 26 species. And most of them belong to the members of the class Bacilli, which is roughly half of it. Alpha proteobacteria, which is another class that you have microbes that can withstand, and even gamma proteobacteria and actinomyceta. This is another big concern, that it can withstand the space conditions. I think Anna, Georgia's student is having a poster about specifically for Acinetobacter thing. Okay, so Phoenix mission 2007 versus metagenomics-based, PMA-based things. Do they persist even after 10 years? These are the novel species that I just told you, which we are describing, and this is the breadth of coverage. So most of them are roughly 1% or below, or 1 or 2 is different. Okay, this is 2016, this is 2018 metagenome. 22 out of 26 novel species present at least 1%. We are all throwing 1% out from our metagenome reads that we have. So maybe we have to come up with a different bioinformatics approach to see whether these are hidden in there, right? Like this. But still it is 1% only. And 40 locations tested, there is no mags of these novel species that we found in this. 22 out of 26 cultured novel species were persisting even after 9 to 11 years when metagenome sequences were mapped. So the average coverage breadth ranged from 0.0045% to 3.91. This is what NASA wants to check. This is what the requirement is based on. You tell me, you are also, you are the experts on the metagenome, how to find this rare microbe. Okay? So in metagenome, the relative abundance showing less, more than 1% only normally considered, and hence we will not be able to detect these rare microbiome that could be easily cultured. That's why we are not able to convince the federal government to use the quantitative PCR-based analysis here, for the unknowns especially. So this is another one. The previous one was Phoenix mission. This is a Mars mission I'm going to show you. And this is, same 182 strains were selected for it. 14 novel species came in, and most of them are, gram positive. Not most of them. All of them are gram positive, I think, except Fingermonas. It's not here. All of them are gram. This is again the help of Karthik and his students. Varsha is here, Pratyay is here, Shobhan is here. So this is based on the whole genome sequencing all those 14 novel species we are describing right now. This is 25 out of 183 isolates, roughly 14%. The previous one was 25%, here is 14%. So further molecular analysis revealed that these novel species these are all novel species from one mission. Not 10 years or 20 years, one mission. Take 100 or 200 strains. If we have gone for 1,000 or 10,000 isolates, that would be more enormous. So, Brevibacillus, Rhinococcaceae, Nocardiaceae, Cellulomonadaceae, and Microbacteriaceae. Okay? So let us look into the same thing, culturomics versus PMA metagenomics. among the 14, only 7 at least having a roughly 1% or so, except 1 or 2, which is more than 10-15%, but we are not getting mags from that as well. Here, 96 locations tested, 164 samples tested, no mags of the novel species, 7 out of 14 cultured novel species were seen when metagenome sequences were mapped. Okay? So these are all spore formers that you are seeing there. The other one is non-spore formers. So do the same calculation. Average coverage breadth is ranged from 0.001 to 16% in some cases, but as the same statement apply here, because we are considering more than 1% are normally considered, the rest we are throwing out. So what are we missing? I'm talking to these guys. They are saying, Venkat, you are doing wrong. You are not considering other things that is in the database. You are relying only on NCBI. How do I know? You guys show me if you are having that. Okay, I collaborated with them, including Karthik and Georgios and then Nikos from JGI. And then they come up with this thing. This is what we have in the DOE database, NGM. IMG metagenome has 127,000 OTUs. And here you have 24,000 mags that is not present in NCBI. And this is the one that you are bringing it. I'm sure your bacteria is going to be in here. So they bragged about it, right? My head is spinning. Why this is not occurred to me? So collaborative things, you don't need to have everything with you. Collaboration will go a long way, okay? So that's what I mean. So March 2020 NASA genome only I tested here. Right now we are expanding to 100 different species that we have characterized by culture are being tested, but this one is based on only those 14 novel species that I'm talking about March 2020. So they have this specific marker based on a whole genome sequence that they have sequenced all the culture and all the type strains of the culture collection. They come up with this novel thing. So only one I'm going to talk to you here, COG13, which is ALAS. So we call them ALAS. So alanine tRNA synthetase is common for everything. So that can be used as a thing with the appropriate 98% CD hit value. Okay, this will give you species level hit. Okay, so they come up with this, Venkat. These are the ones that you are having it. That you are missing, especially bothering one. I'll come to that in a second. So this list, I got it. Most of them are out. And then based on their analysis, they said 10 are out, other 4 are inside. Is it true? So they come up with the word saying most of the NASA genomes are not captured, at least they said. And let's see what they actually came up. They came up with this. These are isolates. I'm talking about isolates because you cannot give a name for the one that is not, in culture, okay? So this is what bothering us. Brevibacillus brevis, which is NRRL type strain, IMGM, which is this portion, and then NCBI, both together here. So that is here. This is a MAG, aeromicrobe. So this is a MAG. This is an undescribed or they have not done further phylogenetic studies to describe this, okay? This is SP. This is still okay. And Sporosarcina SP is still okay. But let us look into the traditional way of doing. What is the traditional way of doing? Most of you know ANI, AAI, POCP analysis. What are they saying? Take the genome. Here you also have genome. Compare one-on-one to see what they are. The one they said it is positive to the Sporosarcina SP. It is only 93% ANA. That means this is wrong identification. And then 93% ANA with cellular mono JPLI that we described, that is also wrong because you need 95% and below. And more importantly, I told you this one 89% only, but we have, they come up with this is the right thing, 98% there. This is the MAG, we don't have the MAG in hand. So we are happy, especially this. Okay, now, this came out. All 14 still not there. So it is not most. None of the NASA genomes are captured by metagenome isolate. Path forward. How do you eliminate the abundant microbe? Because every single project or every single ecosystem is different. If you're doing plant, and what are the things that is actually helping the plant, how do you get rid of the abundant microbes that is junk? Okay? So in our case, what do? We irradiate the samples before we analyze. We take the sample and heat it with the 85 degree, kill all the vegetative cells. If the spores are there, it is there. Or if you're using hydrogen peroxide or plasma or gamma radiation to treat your surface of the spacecraft, use that. Okay, that is one way to remove abundant. But we are looking at the DNA. You want to do the RNA? Yes, you guys are having a fantastic technique to do the RNA, but tell me how much quantity of RNA you need. It is not going to work in this place. Okay? So how do you remove the dead cells? So we were doing this PMA treatment. That is one of the things. There is no one technique to fit all shoes. Okay? This has flaws as well. Okay? PM also has flaws. Deplete the host genome. For example, if you are doing something in plant-related thing, plant genome is going to be 99%. But you are looking for 1%. How do you deplete the plant? So I think Hyagen and other people are coming up with the plant depleting steps before doing it. Likewise, coral microbiome we are doing right now. So we want to deplete those, host. This PMA works best with even human microbiome. You can use the PMA things to remove the host cells. Or develop extraction technology suitable for extremophiles like spores or hard cells, hard cell wall, etc. Okay, so recover both hardies and liable microbes. So split the samples. Do one will go to the bead beating, the other one is not, and then extract the DNA. Or treat with enzymes. We are using metapolyzymes instead of doing this harsh bead beating step. You can use the metapolyzyme. Or here we are in the state-of-the-art thing. Young mind has to put together to tell me, yeah, you have all these things. Let us remove bioinformatically all those, dominant microbes. Okay? Or deep coverage. How deep I can go? Right now I'm going for 1000x. How deep I can go? Long reads. That's one thing that you can think of because these are the dead stuff maybe in fragments, but how long? You are not going to get 1 megabase or 5 megabase of things to get everything done. But whole genome sequence, whole genome amplification, multiple displacement amplification can be done. That is one way to do, but still we are not perfect at this. So use novel bioinformatics tools and approach, remove sequences from dominant microbe, analyze functional genes, don't go only 16S or ITS or something. Rare microbes are spores, psychrophiles, and other things. Actually, you need a phenotype to look for the functional gene. Most of them are, more than 50% are not, you don't know what it is. So NASA-relevant microbes is resistant to radiation desiccation. Those genes can be tapped in. Resolve unclassified dominant level taxa. So that one is more than 90% in extreme environmental samples hidden treasure. We are not, in our case, we are throwing 80% from the Red Sea. And from space station, I'm throwing 40 to 50% of the sequence out because we do not know what it is. That belongs to your rare biosphere or dark matter. Or, we are going to hear soon the next speaker, single cell genomics, but she needs tons of the targeted material to do it. Okay, so we all did this, okay? Why do we need to do this, right? That's another, why you are concentrating mainly on the spores? Why do you worry only that? Because that is the one that survives, I said, right? So with all the translation thing from basic science to enhancing human health, I already told space biology is to support the health of the human there, if you need to live there, and also you need to come up with appropriate pathogen detection and countermeasure development, leveraging this omics data set for medical practice. So I will not have time to go through a lot here. What are the applications of the microbiome research that we are doing here? One of the Kalamiella story, one other speaker here, Georges, is going to talk about this Kalamiella story. And then Klebsiella, we have some of the microbe that is actually acting upon the fungus, so that came from space station, so that you can use that, exploit that and use it. That's producing better in under microgravity. And then prevalence of AMR needs to be done before going. But today I'm going to talk about this particular thing, which is a spore former, the persistent spore former. Anytime you go, clean room, any clean room, any medical facility, I guarantee you can find Bacillus pulmonis, okay? But I'm going to tell the story about face to your face, what that means. So we have genomics, proteomics, metabolomics, and out of the world cream that can be used to protect you from aging. Okay? So I said, keep on saying this word, persistence, right? That is the one you have to be careful. Those are the ones that survive these conditions and be there for a long period of time, correct? First, before that, you have to know what they are. Like, hydrogen peroxide is one of the sterilant that we are using because we are building better, lighter, and faster spacecraft. In Viking days, we built in the 1970s time, we built the spacecraft that landed in Mars in titanium, which is so heavy, you cannot do it now. And also, why we did in titanium? Because there was trying to send it to look live in 1970s in Mars, right? So we took the whole lander and put it into a hot air oven, 116 degree 92 hours. That one step cost $200 million in 1970s. So now you cannot do it because one mission is some mission is $200 million. That one step is $200 million. NASA will not be allowing to do that kind of thing now. So not only that, I said cheaper, titanium is costly, faster, titanium is heavy. So you have to come up with some different material that withstand these conditions, right? And then go to other planet faster. It is right now from 9 months to 6.5 months we are trying to get down so that we can reach to the Mars. For that you are using other non-conventional decontamination procedure, in this case hydrogen peroxide, and we are having vapor hydrogen peroxide. All those is tested, and then B. pumilus are the one that is surviving better than B. subtilis. Okay? Okay, everybody says you have UV resistant 254 nanometer, but Martian UV is 200 nanometer and above. That is more lethal. Yes, more lethal for the tamed puppy, the bacillus that you have, but not the wild wolf. That is isolated from this assembly facility area and also spacecraft surfaces that can withstand 30 minutes of Martian UV conditions that are simulated, and only 2 to 3 log reduction is there. People will say, exposed to more time. That's fine. Everything will get killed. This is not immune to the UV. I'm not saying that. You can kill it. But if you expose it correctly, in 30 minutes, lander is landing, it's shaking off, all the materials outside may go beneath the micron level of soil. If you have a Mars dust devil, then that can block the UV radiation. So this survive those conditions ever if it is under the soil. I'll show you in a second. And also this is fully sequenced. Jill is going to kill me. We've spent $100,000. This was in 2000, early 2000. We did not have that much technology. One bacteria was costed $100,000 during that time. to sequence. Okay, so I said UVC 254. See, this is the American Public Health Association using your drinking water. There is 200 joules per meter square is the standard, and some, bug which is not, ATCC culture, but it's also ATCC culture, but isolated from some environment. That is withstanding a little bit better, but Bacillus pumila for 32, this particular strain isolated from the spacecraft surfaces is actually 1 log reduction even after 2,000 joules. Okay, you got it. But this will get killed in Martian simulation condition, right? That's what they say. So we have this kind of sample holder where you can put a known amount of spores on it, and then put all the other sensors together, and then pull the vacuum to the Mars 6 millibar, and all these things with the UV-vis source, xenon lamp source, you can generate 200 nanometer and then see how that is. So you expose like this. That's what I said. In about 30 minutes, this one only 2 to 3 log, but not the other one. Okay? And if you have the Martian soil, as I said before, if it is sitting underneath the 1 grain of Martian soil, it will be there. This is for 1 day Mars UV condition and desiccation. This is desiccation Mars condition plus UV 1 day. Okay? This one is no Mars UV. This one desiccation Martian UV 1 day. So this and this are same. But if you have desiccation taken from there, it's 3 to 4 log reduction. Okay? Now you have shown simulated condition, everything. Now let's take to the space. You cannot simply take to the space. You need an appropriate, space payload. So this is the space payload where you can generate so many different things. So one is, 110 is the space UV, and then 200 nanometer and above is the Martian UV. You can also pump different gas so that 6 millibar pressure can be made. Here, this is space vacuum. If you put the shield on top of it, it will not anything penetrate, so that means dark. So dark space, dark Mars, space UV, Mars UV, space UV. So these 4 conditions we did. Flow 2008, and then put it on the outside of the space station, which is the, work with the European ESA collaboration. This is what that one, this is where that. So it is exposed for 18 months outside the space station, brought back. See, I told you, some of the, you can have a mirror polished aluminum, but still you have pits and falls. So if it sits on there, that's done. This is ground control 1.5. This is spores after UV are in space for 1.5 years, right? So you have this structure, but we do not know whether it's live or not. So we got all the things then, but we want to double check whether this phenotype is there or not. Some of the things survived, which I showed you, it is because of that attenuation of the UV by shadowing it, right? So this is the one UV space stuff is double the one that, this is the control that kept for 1.5 years. So get killed around 2,000 joules, but this is not. There are spores. The spores that revived again to made spores and did it. That means it is there something, but genotypically, no changes. Phenotypically, there are changes. This one is more important, vegetative cells, because if you want to do some metabolites, what that means, you have to have the live cells. So after Earth return, first generation of vegetative cells isolated from each conditions were exposed, and this particular dark space, 900 joules. I told you 200 joules is the Public Health Association stuff for the vegetative cells, for even spores. This one is vegetative cells of the parent thing, wild type stuff. And this one is the one that exposed and brought back of the same strain, but there are certain differences. The difference are here. This is proteomics. Genomics, no change. Okay? There are certain things upregulated, and what they are, it is written here. taken together, these functional assays demonstrated that the activities of TCA cycles, which it cannot grow faster, right? So TCA cycle and energy metabolism are decreased in space compared to the 1G control. Okay, these are all great. We have patented this bacteria and worked with the drug development people, okay, and then gave the license to them. And then they found something promising in the metabolites, which they are not telling me exactly what metabolite it is, okay? So this is the one we exposed, right? So we made up this particular thing called age-defying serum. It is incredibly expensive because of the name, I think. Name means it is coming from space. So $125 per 30 milliliter serum. What is this, right? So it is actually help restore moisture to your skin and improve visible signs of aging such as fine lines, dark spots, dullness, and wrinkles. This age-defying serum, nothing but hyaluronic acid that you all using it. All the ladies are using, I'm sure. Okay, but this is the one that is actually proven that it is having a better product against the human cell line, and also proven. Here, Madhuri Dixit is using this. So, issues and concerns, which I already told you. So, omic data sets were placed in a NASA Gene Lab. Anybody can freely use it. Some of you are already using it, and I'm sure. And that has a lot of things, not only microbial, that has a lot of other sets that one can use it and test it. This is the help of a lot of postdocs and students, including Karthik students here, and enormous amount of investigators. So questions, and I'm just showing you here when the questions are asked. Go ahead, Karthik.
Oh, any questions from the audience?
This simple video, don't worry. Don't get distracted.
Great talk, Kasturi. So, just a quick word. So, you talked about this pumulus, like Bacillus pumulus, SAFR and ATCC. Means have you looked?
ATCC. Yeah, mine is SAFR.
Yours are ATCC must be the reference one, right?
Yeah.
So, how different they are, like genome-wise? Have you looked into it? I'm sure you guys would have.
We have 10, 15 publications about the genomic difference and the SNPs and all. Even Prathya is looking at with several different strains that went up to coming back to space. The one compared to the ATCC strain, no difference at all. SNPs are there here and there, but it's not big. But proteomic are different. The vegetative cells coming out of that, right? So that is different. These are spores, metabolically inactive, that should not have any kind of problem, right?
There can be like some specific kind of spore coat protein, or maybe some specific mutation in the spore coat protein which is enabling them to become this much rigid.
Absolutely. Whatever you are saying is right. They have almost 10 different institutions has tested, and with the genomes that we have generated, the one that SAFR 32 versus the control strain, we're not seeing that much difference. I can give you the data.
That will be good. Thank you.
Hi, it's Jennifer. I'm sorry, I'm all the way in the back. I was thinking more about the question you asked me yesterday, and I stand by my answer that stable isotope probing is not going to help. But there's potential to do imaging. We do isotope imaging with nanosims, and we've done a lot of spores over the years. I'm not enough of a spore expert, not any spore expert, to know, are there any kind of morphological or physiological differences between a spore that has the ability to, I don't even know the right word, regenerate, come to life, versus a spore that is just defunct? Is there, in your testing, is there any kind of chemical or other kinds of differences?
DPA, dipicolonic acid, is one of the things. when the spore senses some nutrients or amino acids, it starts germinating. So that is DPA, dipiclonic acid. So that is one of the chemical things that people are using it to as a marker to measure as well. But again, when you're handling all those things, you have to start with minimum 10 to the 5 or something. We are dealing with 300 spores per meter square. How am I going to collect 1 meter surface of things where you have only 300 spores are there and remove the dominant microbes? When we are looking at the chemicals, so other things may interfere with your chemical signature as well. Imaging is a great thing, but we have taken 1 meter square surface and putting into a small 13 millimeter filter and look for inside the hybridization kind of thing. Still, I need 10 to the 5 target materials in there in the presence of 10 to the 9 other materials that are not spores. So that is interfering. The images are there, but that also interferes. Somehow we have to remove the dominant microbe. How are we going to do? I'm picking the young brains here. Bioinformatically, they can do something. Bioinformatically, they can remove all this 99% of the microbes that are not relevant, at least to our knowledge, and then look for those only 1% so that we can come up with the appropriate way to make it as a requirement. NASA is not only NASA, other food industries, FDA are willing to go for DNA-based measurement to come up with the requirement, but still it is not happening. I don't know if it is going to happen in my lifetime. But we will do it on a qualitative basis for sure. On a research basis, sure. But is it going to be a kind of federal requirement? We don't know. Yeah, Yamini?
So again, thank you for the organizers. I'm Kane asked some wonderful questions and great talk again. But I wanted, one thing which actually struck me was you said you were looking at spores because they can survive at 80 degrees Celsius in the space flight and the interplanetary distance. I know that the future of space or space biology is to take our microbes and do like biomining, bio-based space agriculture. So how are you expecting, how should that be done? Should we be looking at thermophilic organisms only, or can we explore other organisms or synthetic biology? Yeah, your opinions. Thank you.
A great question. So, number one, right now what we are thinking is everything is happening inside a habitat where human can live. So, inside the habitat where you have a radiation shield and other things, since humans are going to live, it is going to be room temperature. So we are thinking of all the things that can withstand that temperature, temperature-wise. Second, but if you're doing inside resource utilization, for example, you're going to take the lunar soil or Mars soil to make your plants to grow, that is a whole different story about the soil regoliths characteristics. We will be looking at it, what it is going to help us. So that is also going by the side. So we are having regoliths, we cannot get the moon soil, we get only few microgram. So with few microgram, we cannot grow plants, right? So, but we are having an analog soil of the characteristics of most of the characteristics of those things, and then we are testing with various different microbes and other components that can be produced, made in space first. So made in space material mixed with microbes to do however it is. And we are also doing some elevated radiation level which is conducive for human as well. Because we are not going to open up a terraforming now yet. When a terraforming comes, the whole 9 yards is different. When Elon Musk is going and then going dump and somewhere else, there's different. I can say I'm retired, unlike these guys. The Dodge can come back and say it.
Any other questions from the audience?
Yeah.
Enjoyed your talk. Fun talk, seeing your head spinning and the drama of the questions getting resolved. So the question I had was, in terms of the origins of these novel species, so which component of this whole space travel do you think may have led to these novel species? Is it the travel part or the microgravity or the radiation? Are there any thoughts on that?
Yeah, number one, I have not shown the space station microbe right here, so it is not microgravity. Second thing is it's not on the sample that went, collected samples on the probe went and come back. This is preparatory clean room samples as well as surface of the spacecraft before flying. So one of the biggest question here is oligotrophy. Low nutrient conditions is pushing it to withstand and persist there. That's the one thing is driving there to persist, and then it is novel. It is there. It is there in our garden cell, I'm sure, if you go and look for it. Now he's going to look for the things and specifically some of the problematic microbes and develop a diagnostic tool to look for it. Then you can find it. But first you have to find out what you are going to look for it. That's what this one. So clean room, divide off all these junks, but others are not proliferating but thriving. So that is what we were able to get. So oligotrophy is the biggest driver.
Quick question. So how do you get rid of the abundant microbes? Because
that's what I'm depending on you.
No. Through an assay. Because you said you will do some radiation or something like that, but that's going to get rid of the rare microbes as well, right? What am I missing?
Then it is not. When I said rare, so in terms of functionality, that is unique HMO5. If it is dying of that condition, fine. I don't want that. That will not make the red planet green. Nobody's laughing. Is it so funny?
We just have one online question. What would be an ideal spacecraft microbiome?
Obviously, it is not sterile. Basically, it is a good question. NASA never used the word sterile anymore. It's microbial decontamination only we are using that word, okay? What would be an ideal spacecraft microbiome? Ideal spacecraft, basically, if you are looking live, you don't want any microbes. Right now, we are trying to do below detection limit microbiome. So one thing what I have not told you, we planetary protection engineers has to live with today. But astrobiologists who has a luxury of 10 plus 15 years, who can develop technologies when the sample come back. But already the sample is getting contaminated with the Venkat's lab bug. But they can find it. And for that we are actually archiving our subset of the samples and then look for the same technology that you're going to look for it later. Okay? So that's what I say, 10 years back, the oldies like us, we don't, our wives doesn't care whether I'm calling tonight, I'm okay, how are you, good night. But today, if your boyfriend or girlfriend not giving you a call, next day you are not be with them. That kind of technology evolved right now. You have cell phone technology and all those things, right? And the traffic today, maybe tomorrow you will have a sky taxi to come here in 5 minutes. 10 plus 5 years, but not today. So what I'm trying to give you analogy here, as a planetary protection engineer, I have to live with what I have today. Otherwise, the mission will not move forward.
Okay. Let's give a round of applause for Professor Venkat. Thank you for the fascinating lecture.
So hello, it's a pleasure for me to co-chair this session with Mazia, who is a senior research scientist here at IIT Madras, and I'm a faculty in the computer science department. So I'll be introducing the next speaker as she's setting up. So the next speaker is Dr. Tanya Woike. So Dr. Woike is a staff scientist at the DOE Joint Genome Institute. She earned her PhD in microbiology at Eberhard Karls University Tübingen in Germany. She began her role as the microbial genomics program lead in 2009, and in 2021 assumed the position of deputy director of the user program at the JGI. Beyond her JGI responsibilities, she is an adjunct scientist at the Bigelow Laboratory for Ocean Sciences since 2012, and an adjunct professor in the Life and Environmental Sciences Department at the University of California. And she's going to talk about genomics at the intersection of microbial communities and single cells. So we are going to transition from astro scale to single cell scale. Pleasure to have you here.
Thank you. Can you all hear me? Is the mic working? Okay, great. Thank you. All right, so first up, I would like to thank the organizers, Kartik and the other Kartik, for inviting me and giving me the opportunity to speak here today. And my talk today is going to focus on something that I consider sort of the intersection, genomics being at the intersection of microbial communities and single cells. So in the last day, you've seen a lot of amazing talks that are using metagenomic data, that have been capitalizing on metagenome sequence data, and you've seen a lot of great examples on what you can do with metagenomics. My group, within the last 15 years, started to do single-cell sequencing. So that was kind of the early days of single-cell genomics, when Roger Laskin really was able to show that you can sequence the genome of an individual single E. coli cell. And that's when I was super excited and I wanted to try it. It was really hard at the beginning to isolate single cells. Actually, I started with micromanipulation, which I wouldn't recommend. It's really low throughput. But our first single cell genomes were done by micromanipulating single cells under an inverted microscope, as sort of a glass capillary. And then we set up fluorescent activated cell sorting for sort of a workflow where we can use FACS to sort individual cells from environmental samples into 384-well plates. And then we do whole genome amplification and sequencing. And we did that for several years, and we had a lot of fun. But then genome resolved metagenomics came along, and this was really spearheaded by Jill Banfield and her team, and many of you in the room. And it was a real game changer. Suddenly you could extract thousands of genomes from just a couple samples. And not just the number was mind-boggling, but certainly the quality. You heard yesterday from Jill, a lot of these genomes are just top-notch quality. They are manually curated, manually closed. So you can get really good quality data fairly easily and cheaply done, because doing a shotgun library is a lot more straightforward than sorting and amplifying single cells. So we pivoted a little bit, and we started to still use single-cell sequencing, but focusing on more targeted approaches. So for example, we used fluorescent-labeled substrates, cellulose, to pull out cellulose degraders. We used other fluorescent probes where we target cells based on a phenotype or a functional trait that we're interested in, and then we pull them out. Another example you heard actually yesterday in the talk, you heard the word BONCAT, or bioorthogonal non-canonical amino acid tagging, which is an approach that allows you to tag fluorescently label translationally active cells, and then we can also use FACS and sort them out. So it helps you to find some needles in the haystack, for example. But more recently, and that's what I'm going to talk about today, we switched single-cell sequencing to focusing microeukaryotes. And to me, although it's a single-cell genome from this microeukaryote, I really consider these data types mini metagenomes, and I'll show you why. So the premise here is really that a single-cell genome, in that case that of a microeukaryote, can contain other cells inside. So it's really a melting pot of different entities. So you can have your microeukaryote host. It can have food bacteria that are housed in the food vacuole, so they come along. It can have true endosymbionts that are inside intermitochondrial, or there are even intranuclear symbionts that exist. These, in fact, can be infected with bacteriophages. You can have giant viruses infecting the eukaryotic host. You can have virophages, etc. So sequences associated with a single cell can comprise many different entities. And in a way, it's kind of a biological unit. So for us, it's a really cool system to study. So we started this work a few years ago, and we have a few projects ongoing. We're working on single-cell fungi, chytrids, which are early divergent fungi, and protists. And so today I'm going to talk about one project which is focused on protists. And this is led by Frederick Schulz in my group. This is a collaboration with Laura Katz at Smith College. She's a real expert in the eukaryotic portion of the project. And we were really interested in looking at the bacterial sequences, viral, if we could find archaeal, sort of the non-eukaryotic stuff. So in that study, Laura isolated, or her team, her students, isolated a bunch of these microeukaryotes, amoeba and ciliates. So you see some of the pictures here. For example, here, these are Loxodus, these are Halteria. And they isolated them, and they did whole genome amplification and sequenced these. And this is our workflow that we used. We assembled the data, we binned the data, annotated it, and then ID'd any context that looked like their eukaryotic context that Laura analyzed. And then we looked at anything that may be bacterial or viral. And so I'm going to walk you through this because I know there's a lot on the slide, but in a nutshell, from these about 100 protist single-cell genomes, we obtained over 700 metagenome assembled genomes if we binned the data. And on the left-hand side, you see the eukaryotic tree here. On the top are the amoeba, at the bottom are the ciliates. And just to orient yourself, so these branches that are in red are essentially the hosts that Laura isolated the single cells from and that we did the whole genome amplifications from. And then these bar charts on the right-hand side in gray are the numbers of single amplified genomes that we had for each one of these hosts. And then the corresponding MACs and their quality estimates and their taxonomy. So here, in a nutshell, the highest taxon enrichment of these, what we can call protist microbiomes, were in these Halosphenia and the Loxodias. So for example, right here. But I'm going to walk you through a bunch of phylogenetic trees and break it down in a little bit more detail. Okay, so what are these 724 MACs really? So 150 of these, if you put them in phylogenetic trees, they branch with known bacterial endosymbions, so with known symbions of eukaryotes. 258 group with sort of known/putative symbionts, so these are the Petoskey bacteria. 82 with giant viruses, and then we had a bunch that kind of fell within the phylogenetic tree with other free-living bacteria. And I'm just going to talk about these top three groups right here. And I'm going to show you a bunch of trees. So just to orient yourself, these are trees that are either phylum level or class level. We're going to start with the alpha protobacteria. This is just a reference tree. And all the branches that are in red are our metagenome assembled genomes that were associated with some of these microeukaryotes. And which microeukaryote is what you see at the tip of the branches right here. So these kind of colored boxes and circles represent the putative host, so essentially the single-cell genome that they came from. So at the top are the amoeba in square, and then in circle are our ciliates. So starting with the alpha protobacteria, for example, these colored wedges are branches that are composed of known symbiont clades. So starting with the alpha protobacteria, the Rickettsialis and Holospiralis, which you see here and here, we were surprised. We only found them associated with ciliates. So you see a lot of circles at the edges at these terminal tips here. If we then move to the Pseudomonadota tree right here, where we have the Legionelalis and the Coxiolalis, Legionelalis were associated with all the hosts, with the ciliates and the amoeba, and the Coxiolalis only with the ciliates. Chlamydiota also associated with all of the hosts that we find, so you see a lot of colored boxes and circle. And then we had a few MACs that treat together with the Dependentiae, which we also found in both chlamydia, in ciliates and amoeba. And so to summarize that with a few exceptions, which was Megaera and Holospora, most of these symbiont clades had not been found before associated with ciliates. Moving on to the Petoskey bacteria, this is a tree of the Petoskey bacteria. Again, in red are the MACs that we found associated with our microeukaryotes. So you see a lot of red branches. And some of them kind of have, form their own little clades, and a lot of them have orange circles. So a lot of them we found associated with Loxodus, which is kind of cool. So if I would bet my money on something and put efforts into fish, I would probably try and go back and find some of these Loxodus and see if we can actually image some of these symbionts, hopefully true symbionts associated. So here, again, we're covering a high diversity of different Petesky bacteria clades, and the highest diversity associated was with the ciliate, the Loxodus ciliates. And then lastly, we looked at giant viruses. These 82 giant virus mags were found across the nucleoside of Viricoda tree, so this is what you see right here. Diverse giant lineages were mostly found in the Hylosphaenia elegans, which I think are these light green boxes, so you kind of see them dispersed around the tree. And as far as we know, giant viruses have not been associated with ciliates before, at least not in a co-culture system. So now again, this is all sequence-based prediction, but we still think that this gives a glimpse into these microeukaryotes probably having, or that these multi-associations are quite prevalent within these, at least these ciliates and amoeba that we tested. And what you see here is sort of a wrap-up, and this is, these are just bar charts that show these mags on a per-cell level. So each bar chart here is a single amplified genome of a microeukaryote. The colors represent the different mags, or the taxonomies of the different mags. So what we see is a little bit more messy signals. So some of these associations are probably random, or we have food bacteria. But then you see some groups, for example, the Spirostomum right here, that tends to associate with this kind of pink clade, which are our alpha proteobacteria. These are actually the Rickettsialis. So again, this could be a true symbiont association. So again, this would be maybe another candidate where you'd want to go back, pull these cells out, and do some fish analysis. All right, so what's the takeaway so far? To us, this data supports that protists sort of serve as mediator of complex viral and microbial associations. We also think this may be supporting this melting pot hypothesis. Of course, this has yet to be tested, but this particular hypothesis says that protists serve as sort of these evolutionary melting pots for anything that's inside of them. That's been shown for acanthamoeba, for example, in symbionts of acanthamoeba exchanging genetic material, either with each other or also with their host. So that facilitation of genetic change then would, of course, change also the bigger evolutionary trajectory of the host and the inhabitants, the viruses and the symbionts. So what we're doing right now is doing HTT analysis to test that further. All right, now all these, again, all these data is based on sequence-based predictions, right? Single cells are pulled out of the environment, the genomes are amplified, the data is super messy. And then we say, okay, these things may interact or they may associate with each other. But just to show you one example that these associations do exist, and there's of course a great wealth of literature on some of these co-cultures that exist. We had the fortune to have a postdoc, Thomas Tümmel, who's, I guess I would consider him a protistologist, who's really ambitious about cultivation, and I have to say my lab does close to no cultivation at all. But he created a cell enrichment of an amoeba that has an intermitochondrial symbiont. And so we looked at that more closely. So you see here the TEM image of this amoeba. It's a vanilla, so that's the phylogenetic tree of the host. And inside, you can see that they are packed with these bacterial cells. And at the beginning, we didn't know yet what they actually are. We tried to cure the culture of these cells, so did some antibiotic treatment, and actually realized if we try and get rid of these cells, the culture does a lot worse. The growth, at least at the times that we tested it, performed worse. If we then reintroduce the bacterium, the culture recovers, and actually the vanilla grows a lot better. So we wanted to get at the genome of the symbiont, and that also was challenging because when we just did shotgun sequencing, it was super messy, and there was a lot going on. The host genome was probably large and complex, and we couldn't quite piece it together. And it's an enrichment culture, so it's not a real pure culture. So what we decided to do is use flow cytometry because that's something that we know how to do. And we did a two-step sorting process. So first we sorted the amoebal cells, the trophozoites, which you see right here in the sort window. And then we took that sample and we broke the cells open and then relysed them to release anything that's inside. And then after that, we sorted again. And we had 3 sort windows that looked like they could be bacteria. We did whole genome amplification and sequencing. And this P5 sort window right here ended up being hopefully our symbiont, but certainly a symbiont that we think is actually, no, we've now confirmed it with fish. I don't have the fish data here, but it ended up being the symbiont. It's alpha proteobacteria. It belongs to the Rickettsiales. It's actually a close relative to Megaera. It sits right here. So this is the phylogenetic tree of our symbiont. We have a decent genome. It's still in quite a few pieces, 90 contacts, so it's by no means closed. But yeah, so we started analyzing the genome. And here we were looking at secretion system, metabolic potential. There was nothing too exciting. It really compares quite well to its evolutionary relatives. We're also looking at some biosynthetic gene clusters that are encoded. And all this is still work in progress. But this is just to give you an example that these associations really exist. I mean, some of them are probably, even though we predict them with computational single-cell sequence data, which is messy, some of them may not hold up. But you can always go back and bring it in the laboratory, and you can do FISH and imaging and confirm your results. Okay, so the takeaway right here is that enrichment culture revealed this intermitochondrial endosymbiont of an amoeba, of this vanilla. Possibly it's a mutualist. We still probably have to do more experiments to really test that. You can use flow cytometry, or we were able to use flow cytometry and whole genome amplification to give us access to the genome, to be able to sequence that. Not sure what's going on with the light. Okay, speaking of whole genome amplification, I think I have a few minutes left, and I want to spend them talking a bit about whole genome amplification. So I mentioned earlier, we kind of shied away from single-cell sequencing because the genome coverage of single-cell genomes isn't that great. On average, or a median here is less than 40%. That's usually kind of what we see. And again, if you can extract max from environmental samples by the thousands and get pretty complete genome, it's really not worth it to do single-cell sequencing. It's expensive, it's costly, it takes specialized equipment, it's tedious. So why do it? So we kind of, again, shied away from it. However, I also think there are a lot of advantages if you want to look at population heterogeneity, if you want to link mobile genetic elements to your host. It's a great approach. Okay, so this is not good. And this is also kind of just to show you some recent data. Actually, these were recent samples that were processed at the JGI, and you can see there's a great fluctuation in the level of estimated completeness that you get. This is single-cell sequence data, estimated completeness, single cells from different environments. So in soil, we're doing pretty poor. Rhizosphere, phyllosphere, some aquatic environments, we can get pretty good genome completeness. But it's still far from perfect, and in part that's because MDA introduces this coverage bias. This is actually a really nice study from the Chisholm lab where they did Prochlorococcus single-cell genomics. They used an isolate, and they sequenced a whole bunch of the same isolate genomes and just plotted the coverage. And just to illustrate that this coverage bias is not just tremendous, but it's really random. Like it's the same genome, but you get a different coverage bias with each single amplified genome. So a few years ago, actually 2 years ago, Chuck Gavant from Stanford called me up. I knew him from Craig's lab. And he said, hey, we have this new amplification method. Do you want to try this on bacteria? We had tested it on humans, and it works really great. And it's called PTA, Primary Template-Directed Amplification. And so I said, sure, yeah, that sounds great. And I got people at JGI involved. Rex Malmstrom has really helped with this work. And so just to walk you through, at the top here is sort of MDA for people not familiar with it, multiple displacement amplification. You essentially denature your DNA to have it single-stranded. You use random hexamers to prime the reaction. And start amplifying. You have nucleotides, you add a strand displacement polymerase, which is usually phe29 polymerase, and you start your amplification. And the enzyme, it's isothermal, it's not a PCR because the enzyme will just displace, so one strand will get displaced, and then you have a single-stranded DNA again, substrate for primer binding, and this process goes on and on and on. and you get this hyperbranched DNA molecule. So that leads to this coverage in part, and also to chimeras. You get these chimeric junctions. And this whole process then leads, it's an exponential amplification, essentially. So PTA, on the other hand, it's a very similar process, but with the difference that you add terminators. So you make these amplification products shorter. So you essentially cut them short. Again, here is priming, extension, but then you add the terminator, and then once the Phe29 polymerase starts displacing, these short pieces fall off. Now, they are too short to be substrate for primer binding anymore, so you force the primer binding to come back to the original template, right? So you really only amplify off your original template. And this leads, instead of to an exponential amplification, you get this quasi-linear amplification. And, which is really great, you get a lot more even coverage across the genome, which is what you see here. And this was done in human cells. So we collaborated with Chuck to test that on microbial cells, and we wanted to do a benchmark first using known isolates. So we used E. coli, P. putida, and B. subtilis. And we compared this PTA, this new method, to the traditional methods, which is the MDA and WGX, which is essentially just an MDA using a thermostable Phe29 polymerase, with the idea that you can run the isosomal reactions at a higher temperature, and that helps you to displace these high GC templates better. So it works better on high GC genomes. And then, of course, ultimately, we want to test this in environmental samples. So we included an aquatic sample and a soil sample, and for lack of better samples, we picked Berkeley Lab soil, LBNL soil here. So looking at the benchmark data first, these are the coverage plot comparing PTA to MDA to WGX using these benchmark organisms. And these are just some coverage plots, and you can already visually see that the coverage is a lot more even. Now, if you want to be a little more quantitative, we run these Lorenz curves and Gini indices, and this is what you see on the right side. So these dotted lines, if there was no bias in the data, you would get this dotted line, right, if the coverage was perfect. So the closer these Lorenz curves are to the dotted line, the less bias you have. So you see the turquoise line is pretty close, so these Lorenz curves look pretty good, and then you could also convert that into Gini indices. The lower the bias. So again, our PTA has much lower bias than MDA and WGX. Now, what does it look like in terms of the genome that you can recover? For E. coli, we got well over 90, 95% of the genome. For B. subtilis, almost the complete genome. Now, these are not closed. They're still in many pieces, but you get a lot of the genome back with PTA. For P. putida, not so much, but I do want to say this was at 1 million reads. And if we add more data, so we can run this at 4 million reads, that's what it looks like. So suddenly you get more than 90% of the genome back. So that's a huge improvement, I would say. We then also looked at mismatches, because we want to make sure we're not introducing some sort of errors now. And the mismatches are largely on par with the other whole genome amplification methods. And as far as other general genome assembly statistics, we look at contact length, contact N50, et cetera. It's about the same. There's no improvement, but it also doesn't look really worse. So these genomes are still fragmented, but you get the gene content is there. Okay, now what does it look like for environmental samples? That's what we're really after. And these are assemblies at 1 million reads. So for our water samples, the median estimated completeness is at over 80%, and again, this is at fairly low sequence, like 1 million read for a single amplified genome isn't all that much sequence data. For soil, it doesn't look that great. Soil is difficult. There's a lot of stuff in soil, so single cell sequencing has always been challenging in soil. As far as contamination, beware that these PTA reagents may not be super pure. We're working with BioScribe, the company that sells them, and they're really actively working on cleaning them up, but you will find some human DNA contamination as well as bacterial contamination in these kits. And then lastly, assembly statistics, of course, are much better, or the MESAC standards here, you get a lot more medium and high quality genomes compared to PTA and MD and WGX. We also looked at mobile genetic elements. That's something I think where single cells are quite good at because you can link them. And we've looked at studies in the past where we took the same sample, we split it in two, we did shotgun metagenomics and single cell sequencing, and we were able to show that in the single cell genomes, you find a lot of mobile genetic elements that are not binned in the metagenome assembled genome. So we here looked at phages, and we also looked at plasmids, and you can see that we can detect in the PTA ZACs, we detect a lot more. Also, we have a lot more assembly, but there's a much higher fraction of ZACs with viruses and with plasmids. And lastly, we looked at biosynthetic gene clusters, which we can also recover a lot more with the PTA-amplified single-cell genomes. Lastly, we looked at taxonomic classifications, and for PTA, as to be expected too, we need less data to do proper taxonomic assignment. So what we're doing next is we're adopting this PTA to these semi-permeable capsules. It's a really cool system. The company that sells these is Atrandi, and they essentially developed these capsules, which are little reaction vesicles. They are semi-permeable, so you can encapsulate single cells, really tens of thousands. It's pretty high throughput and pretty quick and easy, and you can have tens of thousands of them in a tube. And then essentially, you have these tiny little reaction vessels that allow you to do multi-step workflows, right? You can, the cell, you can lyse it, and the high molecular weight will be retained within the semi-permeable capsules, but the reagents will flow in and out. So you can do lysis, you can do whole genome amplification, or whatever chemistry you want to do. So we started using these, and this is kind of very rough data using some E. coli isolates, but here also we compared MDA to WPTA, to PTA in these semi-permeable capsules for single cells, and we can recover close to the complete genome of these E. coli in these vesicles. Now, if you look at MDA, it actually looks really good too, and we think it's because the reaction volumes are so small, the bias is a lot smaller. So the overall assemblies are pretty good. So the takeaway here is primary template-directed amplification provides really a big improvement to the traditional methods, MD and WGX, that we've used for the last 15 years. And I think in conjunction with more scalable approaches, this will continue to make microbial single-cell genomics a pretty valuable complement to metagenomics. I think it's still, of course, not at the scale as to what you can do with metagenomics, and it requires more specialized equipment. But there are some questions that you could ask with it, and specifically, again, linking mobile genetic elements to hosts. Maybe Jill is interested in using it for the Borgs and linking it to their hosts in a high throughput manner. So we're pretty excited. So we're going to start doing more single-cell sequencing again. Now, my second hat at the JGI is that I'm the deputy director for the user program, so I can't not show the slide. I just want to bring to your attention, we have these resource calls. We are a Department of Energy funded user facility, so we provide sequencing, metabolomics, and DNA synthesis to the community, to researchers around the world for free. This is proposal-based, so you have to submit a proposal, and the top proposals get accepted, and we do the work for you for free. Just the work needs to be within the DOE mission area of bioenergy, carbon cycling, biogeochemistry, and bioproducts. We have a lot of calls that are open right now. In fact, our large-scale call, you can request up to 10 terabases of data, so that's a lot of data. We can do single-cell sequencing for you. We do the flow cytometry. You heard from Jennifer yesterday. We do zip metagenomics. We do the fractionation for you. For the metagenomics, even we take projects that are larger than 10 terabases. If you have large metagenome projects specifically, so you heard from Kartik on the Lake Mendota time series data set. So that's a perfect example on what can be done. And it's great to see what kind of cool science can come out of these time series data sets. So with that, I'm going to close with acknowledgments. These are people in my group and other staff at the JGI. Rex is a really great collaborator on all the single cell work. Laura's team and her students were involved in the proto single cell work, Chuck in the PTA work, Iva in the cultivation and funding agency, our Department of Energy, and Templeton Foundation. And with that, I'm happy to take questions.
Thanks. Thanks so much for the interesting talk. Yeah, so questions? You can go here, I can go there. You can ask. Yeah.
Go first. Hi, great talk. I have actually two questions. One is a technical one, which is that do you quantify the abundances of the MAGs from the protist sequencing? Do you then go back and take a pool of protists and do metagenomics on that, just the vanilla metagenomics, and then use that to quantify the abundance
Yeah,
of the MAGs?
we have not. No,
Okay.
we have not. Just these samples are pretty complex. We haven't done that, no.
Okay. And I mean, related question, what do you do for control in there? How do you ensure that the MAGs are not from some sort of contaminant coming out of whatever?
Yeah, for the controls, that's a great one. We think about controls a lot, and it's a hard one for the single cell work. So we do our best to have a clean workflow as much as possible. For the single cell protist work, they're being washed. They're big enough that they can be manipulated under dissection scope, so they go through washes of sterile buffer solutions to clean them as much as possible, but we cannot, and that's why I'm saying these are sequence-based predictions, right? We cannot say for sure that, these are symbions, or I think, for us, this is more hypothesis generation, and then we would encourage other people, or for some of these cases, we may go back in the field, and then come back with the samples, either try enrichment cultures or try to do FISH or other microscopy imaging.
I guess I was thinking that perhaps the metagenomics idea that I suggested with many different samples could help you correlate the abundance of the host with
that could be an additional control,
something.
yeah.
Okay, one last question, sorry. It's a controversial one. Have you seen some of the cancer microbiome papers that have come out?
Yes.
So many of them claim that these bacteria are intracellular to cancer cells. Yeah, and these are diverse, often gut bacteria, like Bacteroides, Firmicutes, Bifidobacteria and so on, which have never been described as being intracellular with eukaryotic cells, right? Symbionts, I guess, staying for long periods of time, right? What do you think about this?
Well, so, at least for the environmental microeukaryotes, we do, for the vanilla culture that I showed, we do have image data to show that these are, in that case, intramitochondrial symbions. There are other studies that have shown, even intranuclear symbions that exist through imaging. I can't speak so much for the gut studies.
But have you heard of people use similar approaches for cancer cells and see if they can convince themselves these are actually intracellular?
Maybe, I don't know.
Okay.
Yeah.
Hi, here.
Yes.
Two related questions. One, is it possible to use the gene content of for the first part of your talk, for the protist work, is it possible to use the gene content to be able to say something about predicted either mutualistic or symbiotic relationships between hosts or between phage bacterial combinations and so on?
Yes, so we started to do that. In fact, we have a project, I didn't show that today, but another project, we're using the sequence data actually started out with known symbions of microeukaryotes and built a machine learning classifier to predict lifestyle and look at actually which features are most predictive for being obligate versus facultative and so on along the spectrum. So that's a big study where we took all public MAGs and built this classifier and then trained it and looked at it globally on the scale of a bacterial archaeal tree. And actually that study showed, at least, again, this is also just sequence-based prediction, but it predicted that actually 10% of all the MAGs were predicted to be symbions.
That's cool. And on the phylogeny itself of the protists and the putative symbionts that you showed, do you see cases where the same or similar-ish looking symbiont is shared across multiple groups in your phylogeny?
In the sample, yeah.
Yeah,
Yes.
and potentially sort of saying, okay, here are switches either in terms of host or switches in terms of lifestyle as different hosts perhaps capture different symbionts and so on? Or is that level of detail not possible to get given the relatively low completeness of the genomes?
I would say that, as you saw in the chart, a lot of these are low What we could consider based on the standards, low quality genomes. We have some medium and high quality, so I'm not sure we can do that based on the quality. Yeah. But again, if we now switch, so we started running our single cell microeukaryotes through PTA, and we haven't analyzed the data yet, but we're expecting much better genome completeness, so maybe that's possible using that data. Yeah, great question.
Okay, Tanya, this is Venkat. Good talk. So in one protease, you have got some 700 mags or something. So which cell you wanted to sort? How do you know which one you are going to sort? In other words, the other question is, if one of the mag is extremely low, what is your limit of detection in pulling it out from that particular sample?
Yeah, so these over 700 mags were spread across all the 100 single-cell microeukaryotes that were isolated. These were isolated purely based on morphology, and the CUTS lab saying, okay, we're interested in Halteria, we're interested in Hylosphaenia. So she knows what she was interested in, and she manually pulled them out for whole genome amplification.
So it is not a kind of limit of detection that you will say, based on qPCR after you get it, then you will come to know how much is there. Because what I'm saying is, can I apply that for spore detection?
For spore
You don't need to answer now because it's in the middle of some break now. We can talk later.
Okay.
So let's thank Dr. Tanya again. Yes, I guess we have a break for half hour. Okay, half hour break. Yeah, 11:35 we'll try to come back. This concludes. Yes.
laser pointer. Let me try to. I think.
Is it not working? This is supposed to be the laser pointer. One second.
Is it turned on from the side?
It's connected only. Yeah,
No,
it's
no, no.
connected.
Is it turned on?
Yeah, it's on.
Oh, did the battery go?
It's on only. Huh, okay.
Okay.
Yes. Is that okay?
But
yeah,
red
this
one.
This one.
Red one.
Yeah.
And I suppose this will also move the slides back.
No,
yeah.
I don't think that
But
works.
other people use a green laser. Green laser is more easier to see.
One green or?
It's yours. Okay. Then no problem.
Yes, I'll first introduce him too, and then I'll do this too.
Oh my God.
Okay,
Perfect. Thank you.
Right, so we'll start with our next session, Space and Built Environment Microbiomes, and this will be chaired by Dr. Revati Krishnamurthy and Dr. Varun Agarwala. So Dr. Revati Krishnamurthy is a molecular microbiologist by training. Her primary interest is in understanding the biology of bacteriophages, viral predators of bacteria that exist in environments ranging from bathypelagic zone of the ocean to the human gastrointestinal tract. Currently, she's a research scientist at Quadram Institute of Biosciences, UK, working with Dr. Evelyn Adrianson's group. Her field of work now is investigating the virome of early human gut using samples from the longitudinal Pearl study. Also, Dr. Varun Agarwala, he's an assistant professor and principal investigator of Microbiome Therapeutics Lab at Geo Institute. He performed his doctoral research at Perelman School of Medicine of University of Pennsylvania, focusing on problems related to personalized medicine and genetics. His lab focuses on engineering of complex microbial communities across body sites like gut, skin, vaginal tract, using combinatorial approaches such as defined FMT and personalized probiotics to target health and disease. So over to the chairs.
Okay, so thank you for the kind introduction. I am very pleased to introduce Dr. Fumito Maruyama.
Yes.
Perfect, I didn't screw up. So Dr. Maruyama is currently a professor at the Microbial Genomics and Ecology Laboratory, the IDEC Institute at the Hiroshima University in Japan, as well as an ad honorem professor at the La Frontera University, Chile. He graduated from Osaka University, Japan, with a focus on microbial ecology and environmental microbiology. Dr. Maruyama's research expertise spans environmental microbiology with a particular focus on bioaerosols and microbial dynamics in water supply systems and built environments. A notable strength of his research is the ability to perform high precision qualitative and quantitative analysis on low biomass samples. My God, this is of so much relevance to humans too. Particularly excelling in culture-independent single-cell detection techniques. And the title of his talk is Unveiling the Hidden Dynamics of Non-Tuberculosis Mycobacteria in Built Environments. Looking forward to it. Thank you.
Okay, thank you for your kind introduction. Before I start, I'd like to thank you all the committee members to have this opportunity to speak in my presentation in front of you. Today, I'd like to talk about this title, but actually, my laboratory has a lot of research topics, so I will introduce a little bit firstly about my laboratory. So my laboratory just... So, Michael, I use here, this one. Where? It's work? A little bit. That's better? Okay. Okay, can you hear? Okay. So, firstly, I'd like to introduce my laboratory. If you have an internet connection, please just type MGE and Hiroshima, then you'll find my laboratory homepage is like this. So my laboratory just started from 2019 after my movement to Hiroshima University from Kyoto University. So previously I belonging to Kyoto University, Graduate School of Medicine, but actually I'm belonging to a lot of different kind of schools, including natural science, general education, and dental science, pharmaceutical sciences. So it means I belong to a lot of different disciplinary sections, faculties. So one of my research line is related to bioaerosol after 2017. So this one is before COVID-19. So that means after COVID-19 condition, so many people look my review related to this kind of transmission of airborne bacteria across built environments. So recently I published this kind of review related to bioaerosol, but focus is resistome in freshwater bioaerosol and their impact on drinking water. So after that, one of my collaborators in Spain published this paper in PNAS, maybe very 3 months ago. After that, just when published, I wrote a commentary for this paper. And then after that, very recently, a scientific news reports the importance of these topics. So it means as introduction, my focus is related to low biomass environment and also related to bioaerosol and resistome and fresh water, especially focusing on drinking water. So to perform this kind of topics, I have many collaboration with foreign countries, but about these topics, today's topics, main collaborator is from France, CNRS, and IS Global in Spain, and also have a collaborator in US, in Harvard. So this kind of topics need planetary health approaches. Maybe most of you know about the One Health, originally from the Global Health, but recently many people know about the Sustainable Developmental Goals determined by United Nations, and such kind of thing we need to consider in addition to One Health. That's why planetary health concept recently appeared and growing rapidly. So that's why now I try to create some international collaboration network called IBSE, that is International Bioaerosol and Research and Water Research Center. So now I'm just struggling to get the funds to create this network, but before that, I need preliminary result to get some funds. So that's why I will talk about these topics to you to attract you to join our project. So, so as I said, I have collaborated with France, Chile, and Spain, and the US. And in my laboratory case, I have a lot of devices related to this topic. This is just an example. So if you contact to access to my laboratory homepage, you can see all of my laboratory equipment. These are my own laboratory device, so which includes, for example, clean room we can use freely, and also my laboratory has this kind of custom bioaerosol chamber and microfluidic device to separate the eukaryote from bacteria without any staining. I mean, the label-free separation of specific microorganisms is possible. And also, my laboratory has a laser confocal scanning microscopy and also image flow cytometer. And also, I have collaborated with private company to monitor the bioaerosol abundance in the air in real time. This one is also real-time viral counting device. And also for this today's topic, important device is this kind of suitcase laboratory. This suitcase includes many different kinds of small devices, including beads beater and also qPCR and also nanopore sequencer and qubit to count the DNA quantity on site. So this is clean bench, clean booth. This is custom one, and all of this can included in this large suitcase. Unfortunately, the weight of this suitcase is 30 kilograms. But so now I move to today's real topics about NTM. NTM is a non-tuberculosis mycobacterium. Maybe all of you know about tuberculosis. Tuberculosis is so famous. But in Japan, now big problem is caused by non-tuberculosis mycobacteria. Usually I say NTM here. So as you see, the occurrence of NTM per population is growing rapidly after 2000. So many people, many researchers, medical researchers hope to understand and clarify what is the reason for this. So especially only in Japan, this kind of trend we can observe. So that's why this is Japanese, but this is a NIH Japan priority list for AMR, antibiotic resistance bacteria. So here says Acinetobacter and Pseudomonas and Enterobacter. But important thing is here we can see NTM. And this is CDC and WHO standard. And then CDC and WHO says this red part is very important. But only in Japan, in our priority list, we include the NTM. So that's why maybe this is a rare case. But in Japan, this NTM is very, very big problem and emergent problem to be solved. So until now, we already published several papers related to NTM, and also based on our paper reference researches, we found more than the mycobacterium NTM, especially for avium. Avium is a major species for NTM infection. Avium can found almost everywhere in the natural environment, including pig and soy and cow and plant, and also river and our drinking water and tap water and our bathroom. But important point is maybe you know if someone already been to Japan, Japanese culture is like a bath culture, so we can find a lot of hot springs all around Japan, and then especially for the older women, is more than older women. I mean, around more than 60 years old, is a patient for this NTM infection. So some medical doctor says. Japanese women usually really love to take a bath, and they usually take a bath, not shower, every day, and they use usually around 1 hour every day. So it means medical doctor believe the source of infection of this pathogen have to be Japanese bathtub. Actually, based on the strain typing based on viable number of tandem repeat. We say VNTR, and also whole genomics SNP analysis suggests. We found the patients, isolates from patients and isolates from bathtub is the same. But this is one of the preliminary results I published previously. Based on this kind of whole genomic analysis and VNTR analysis, Mycobacterium avium can found everywhere in the natural environment, including Japanese soil, and also we can find many Mycobacterium avium in the world, including US case. So if we try to identify which genotype can infect to human, we found only Bastard type can infect to can or identical to the isolates from patients. So it means even though we found a lot of Mycobacterium avium in natural environment, our infection source have to be related to our bathtub or related to environment. So that's why to identify the infectious source of NTM, I check the references firstly, what kind of approaches can be done. Then one of the famous paper related to my field is this paper reported in MBO, and this is reported by Matthew. And then when I asked him why you published this important paper to MBO, maybe I believe this one can be published in better journal. But important limitation for this paper is problem in transportation, water transportation. Because as you know, many people know microbiome in a natural environmental sample can be changed very, very rapidly, but how much affect to influence on the community level by transportation is not so known. So that's why my case, I check the effect of transportation on microbial community by taking a sample from different area in Japan, like this. This is a water source. We can drink microbiome in water by on-site analysis of microbiome by suitcase laboratory, and we send a sample with cold temperature and or normal room temperature. So it takes 1 to 4 days after sampling to our laboratory. So maybe you know, maybe you love sushi, I believe so. And then in Japanese cases, we have a very, very good cold chain to send sashimi to everywhere all over Japan. But so if we check the monitor the temperature for during the cold chain, I found really Japanese cold chain is very nice, and during transportation, temperature is very, very stable. So this is the result. So when I quantify the bacteria, this is the on-site number, but after 1 day, even if we use a cold chain, the number is 70 times more. And if we transfer the water, even 1 day, the abundance is 5,000 times much more. So it means that abundance is totally different on site and after transportation. And then, for example, this is a result of community. So as you see, here is on-site, and this is cold chain, cold transportation, and room temperature transportation. So as you see here, proteobacteria increase a lot, and these are not so many compared to proteobacteria, and usually almost expect proteobacteria decreased rapidly like this. So it's including pathobacteria. Maybe some of you are very interested in pathobacteria. So it means that to analyze the pathobacteria, you need to fix or analyze the pathobacteria on site immediately after sampling. So this is the same for mycobacteria, but here I cannot see, because the number of mycobacteria in raw water is not so much. So, but anyway, community structure changes rapidly, and also Shannon index is becoming much lower than on-site, even we use cold chains. And this is, so using this kind of knowledge, we now doing the drinking water sampling every 3 months at 4 sites. So every 3 months now for more than 3 years, I take a sample from the river, groundwater, and go to water treatment plants, and then take a sample, and also we take a sample from volunteer house. So before that, this will be very interesting for Pathobacteria larvae. So as you see here, so our source of drinking water has a lot of Pathobacteria. So for example here, you can see the 44 or around 60% of bacteria in our low river water, groundwater is include a lot of Pathobacteria, and the number of Pathobacteria OT ASV is around 400 so 10% of ASV is from Pathobacteria. So I think this is very interesting for many Pathobacteria larvae. And then maybe if I think some of you want to get this water, then I recommend you to buy this sake. This sake is made from this water. You can obtain even in US very easily. Just you need to buy sake. Okay, then this is a result of Mycobacterium after water treatment. So this is raw water, and after sand filtration, and after final chlorination is here. So this is different IBSE water treatment plant. So you see here, you can find so many Mycobacterium after chlorination. So, but important thing is after chlorination, Mycobacterium become abundant, but after from water plant to end point, I mean the outlet of the water plant system, maybe end of the normal people water normal house after the end point. So it means from fresh water to end point, it takes stagnation time is around 1 day. Then after 1 day, not Mycobacterium, instead Phyllobacter become abundant. So it means Mycobacterium can be killed by chlorination, but it takes at least around 1 day. So maybe in Japanese cases, as you know, we can drink tap water everywhere in the world, around Japan, no exception. Even in the park or anywhere, you can drink tap water directly without any poisoning. So it appears Japanese water is very safe, but it is better for us to drink water after 1 day after chlorination. So this is just a correlation analysis using different kind of physical chemical parameters. So this analysis includes pH and turbidity and total carbon or temperature like that. But anyway, Mycobacterium can be abundant and highly related to chlorine treatment. So maybe you know abundance of NTM in US and EU is around 50 times more different, because if we use a qPCR or relative abundances in US or EU, around 20% of total community in tap water is Mycobacterium. But in Japanese cases, we found around only 0.3% of community is Mycobacteria. So maybe chlorination will be the major reason for the difference between countries. But anyway, we try to isolate the NTM, especially avium, from the volunteer house. So this is just a simple trial with obtain the water, try to isolate the NTM from volunteer house. So this is just a simple result, but important thing for me is that if we found TM, like this is orange is the NTM curve. Detection rate for avium also high when we found more NTM. So it means not like other pathogen. I mean the normal pathogen usually different species can compete and usually has an inverse relationship in abundance. But in the case of NTM, usually NTM can coexist. So it means more number we found NTM in one place, we found more species in the sample. So it's for me, it's ecologically very, very interesting. But anyway, this is just an ongoing project for me. That is to identify the infection source of NTM. So now we try to collect the sample from many areas, which includes this time I won't talk about, I already had a lot of isolates in the region, and also I had a clinical information I will show you later, and I have a bathtub isolates, and also I won't talk this time, but I had several isolates from NTM isolates from aerosol, and also as you see, I have a lot of isolates from water supply. So I think if we combine this kind of data set, and after that I will do some genomic analysis at the SNP level, then I can identify if the water system in Japan can be a source of infection or not. So next topic is related to geographic information system. Because this kind of approach is not so much, but some of researchers use this kind of approaches using epidemiological data set. So this is one is not our data, but this suggests, depending on species difference in NTM, there is some locality, and this case intracellular occurrence in west side of Japan is higher, but the avium case not so much and even distribution. So this kind of thing is interesting for me because I am microbial ecologist. So that's why I obtain the patient information from hospital and then try to combine with geographic information data set. So this, because some my colleague says, especially for the intracellular, not avium, can fly from China to Japan. This is one of the publication 2020, but I don't think so. So that's why I put the information, patient address to this kind of map, and then I found this kind of red area where is a high occurrence of NTM infection. Here is the most populated, crowded area, but the frequency is not so high. So these regions' characteristic is they use the same water system, or this is like a basin. I mean, this area is between mountains, so maybe aerosol will be key. So that's why, but this is not enough number, only I use 130. So now I obtain the 1,500 cases. So now I doing some this kind of same approaches to clarify, is there any locality in different species? And also, after these kind of year, if we can see some difference between 2000 and after 2012. But recently, I need I get more data from other places. So now this is an ongoing project, but I think this will be a very interesting approach for you. So Another thing is, this is very interesting, more interesting for me is this data from soil. I use a 2,000 soil sample data set from this kind of different kinds of soil, which includes a field, forest, grassland, orchard, patty soil, and sandy beach, for example. So using this big data set, I analyzed the top 20 ASV related to human pathogen. So I will give you one example of this analysis because maybe you remember, still remember about intracellular cellulite. So we found that intracellular in this soil big data sample data set, and then distribution is like this if we use a natural environmental sample from soil. So as I said, some other researchers reported the west side of Japan is high occurrence of intracellular infection, and this is from patients' data set, not from real natural environmental sets. So as you see, if we use a natural environmental set, maybe around here will be the higher than west. So it means there is some difference between patients and the clinical information and the environmental data sets. So, and also we found intracellular especially high, exist high ratio especially in paddy field. So this kind of information no one knows about that, especially medical doctors don't know this. So for me, my interest is what is the difference between clinical and environmental isolates. And so, but only genomic information we cannot find because still we have a lot of hypothetical genes in avium genome. So maybe, you know, 1/3 of essential gene for E. coli is still hypothetical, as you know. So in the case of avium genome, there are so many hypothetical genes still. So because only tuberculosis genome has a lot of information, but for avium genome, not so many experimental evidences exist. So, but anyway, I have, this is a TN-seq result, transposon mutagenesis sequencing result. I found the environmental isolates, I mean bathtub isolates, and clinical isolates has a very different in the essential genes. So maybe this kind of difference in gene have to be the key for clarify the difference in the distribution in this kind of things. So, but anyway, this is summary, but I don't need to explain and enough time. So anyway, thank you for listening, and please join my project if you have interest. Thank you so much.
Thank you. That was a wonderful talk. In the interest of time, let us limit ourselves to just one question. Yes.
Fantastic talk. Can you tell us more about the Pateski bacteria? Could you find any associations? Because usually they are they have an obligate relationship with other bacteria, right?
Yes, that's a good point. So maybe this water is from ground, and as you know, in Japan there are a lot of different kind of hot springs and groundwaters because you know Japan has a lot of earthquake, so it means that there are so many different plates can exist. It means mineral or metal in each plate is different, and a lot of volcano also. So it means depending on the region and water, the microbiome is totally different. So, but in this case, very strange thing is we collect we have a shotgun genomic data for this one also, but still not analyzed. Maybe I need to ask to JGI, for example. But anyway, I filtered this sample to 0.2, not so below. And also, I collect the below 0.2 fraction, but below 0.2 fraction don't have much Patis bacteria, only about 0.2. So it means some relationship with maybe, I don't know, fungi or algae or some archaea, but this water don't have much archaea, very few percentages. But anyway, now I hope to analyze the short-range genomic data and hope to make a good fish flow for the microscopic analysis. So, but anyway, not yet. Sorry for that.
Thank you. Maybe let me squeeze in.
But
I mean, this is relevant for surveillance, right?
Yeah. One reason is we take this from the natural source. So natural source means like avian or borobic water. So it means no filtration. So it means copy number is like this number. So this number is not so much higher than compared to tap water. Tap water usually has only 10 to 4. So originally high number, and natural water means there are still some nutrient for the bacteria.
No, got it. Thank you. Thank you so much. Thank you, Dr. Maruyama. And yeah.
Thank you.
That was a wonderful talk from Dr. Maruyama, which says that why we have to study what we study in a One Health approach way. So our next speaker for the session is Dr. Georgios Miliatis, and he's a lecturer in the discipline of bacteriology at the University of Galway, Ireland, and he's a member of the Antimicrobial Resistance and Microbial Ecology Group and at the Center for One Health. His research focuses on 3 main thematic areas: applying a One Health approach to combat antimicrobial resistance, and understanding the effects of spaceflight on microorganisms, and exploring the links between the human microbiome and mental health disorders. He's also interested in examining and understanding non-trivial quantum phenomena under physiological conditions. I'm so excited to hear from you, Dr. Georgios, and he's going to share with us today the new horizons in One Health using a curious case of Calamilla pearsonii. Am I right? Yeah, thank you. Over to you. No.
No? Yeah, okay. Right. First of all, thank you so much for the invitation to be here today. I'm very, very happy. It's my first time in India. And so thank you very much to Karthik and Karthik for inviting me. So we came all the way from Ireland, and we're very excited to be here today. I'm here with my PhD student, Anna, as well, who has a poster. So the talk today relates to new horizons in One Health, and for that I use an example of the curious case of Calamilla pearsonii, which we've been studying now for a couple of years in collaboration with Venkat and other people that are present here today. So I am an assistant professor at the Center of One Health at the School of Medicine at the University of Galway. So we approach spaceflight from more a human perspective, from more of an infectious disease perspective. So that's the kind of realm we're trying to map. So a few words for where we're coming from. We're coming from the University of Galway, as I said, which is kind of based in a medieval city on the west side of Ireland. It's quite a long way from here, but it's a beautiful place to visit if you ever in Ireland. And our group, it's a small group. We're 13 to 14 people overall in the discipline, and we're located in the School of Medicine, which is based on the Riverside campus of the university. So our research focuses on infectious diseases. We are part of the School of Medicine. We're very medically oriented, antimicrobial resistance, and as of recently, we are trying to kind of use quantum algorithms, quantum computing that is coming up to bypass a lot of the limitations we have in bioinformatics, because I'm a bioinformatician by trade. We're very excited about this new development, but it's still on the very early stages. So let's start off. I just want to pinpoint before we go more into space and so on. I just want to pinpoint, so we're on the same page, what's the concept of One Health and why it matters. So the concept of One Health is very simple, but it's very powerful. It's a very basic principle that human, animal, environmental health are linked to each other. So we cannot see our health as independent of that of our environment and the animals we share the environment with. And we had a very recent example, the case of COVID-19 pandemic, which was due to a zoonotic pathogen. And 75% or more of infectious diseases are zoonotic in origin. So this is an example of why we need a One Health approach. So One Health requires multidisciplinary collaborative approach, as most science nowadays does anyway. But One Health has been applied in policy quite a lot. So mostly in relation to tackling antimicrobial resistance. So we have a global action plan on antimicrobial resistance by WHO, which is based on One Health. In the European Union, we have a European One Health action plan, also against AMR. In Ireland, and our research directly translating to the national plan in Ireland. We're now on the second iteration of our One Health action plan on antimicrobial resistance. So we're trying to tackle antimicrobial resistance and other zoonotic diseases from all angles, from environment, animal, and human. So One Health matters nowadays more than ever because people live closer together. We have changes in climate, in land use, more global travel and trade, and animals are nowadays more than just food. So we live much closer to animals. Sorry. But also overall, if we were to summarize it in one sentence, our connection to our environment has changed. And it has changed so much so that the edge of the anthroposphere, and by anthroposphere we define the outmost limit human activity can impact, has expanded, and is expanding beyond our planet. Okay? So now we have, Venkat gave an excellent talk earlier, we have rovers into multiple celestial bodies. We send people up and down to the International Space Station all the time. I don't know if you're aware, but 2023 was had a record number of space flights. So there's a lot of activity in space. And as we explore beyond Earth, we inevitably introduce our microbiomes to new environments. So the first thing we did is we started thinking about how spaceflight and celestial body habitation can have a link with infectious diseases and how these two can be merged together or might be merged together. So we sat down with infectious disease experts. Some of them were behind the national action plans on COVID-19, for example, so very experienced people. So we came up with this diagram as a high-level depiction of the interactions between spaceflight and infection risk, public health, and so on. So it's a very simplified diagram, but practically what it suggests is that when you're in a spaceflight environment, if there is an increased infection risk, it's coming from changes on your own microbiome, which were well evident in spaceflight, for example. It could come from changes on the microbiome of the crew. It could come from the spaceflight environment. So that's why it's very important to define what potential pathogens can survive during spaceflight, because not all can. And changes to host susceptibility, which is very well evident as well. So human immune system is not functioning as it should in microgravity conditions, and this seems to be irreversible. So as of for the duration of the space flight, the immune deficiency remains and is only reverted upon return to Earth. But then also we started thinking about what happens to these microorganisms that may go up there, change, and maybe accidentally reintroduced back to Earth. What does it mean for public health on Earth, and what would that mean for Earth's ecosystem? And then we have the second scenario, which is hypothetical at the moment. We don't have any celestial body habitats, but one day we might, and we might be alive to see that. And again, the stressors there are different. They would be different and unique, but you would have host susceptibility that would be affected, and you would have altered terrestrial microorganisms that they would be introduced to this new environment, and they would change, and they would adapt, and they would be, they might become something different to what we're used to. And also, you may have extant microbial life, if present, if any day we found some, and then you would have interactions between the two that they're unknown. So this is practically an overall arching theme. But in a very simple sentence, what I would say is that in space, our epidemiological matrices don't apply the same way. Okay? So think of it from a case of COVID-19. We say to people, stay 2 meters apart. That was a national guideline. That's because we know droplets can travel up to 2 meters. Right? In space, this is not true because you don't have gravity. So what is the R0 values? What are the epidemiological values for human respiratory pathogens, other pathogens during spaceflight? Or how do you protect people in spaceflight from infectious diseases? And now that's not a problem so much because we're sending up healthy people, they're well checked, they're isolated before they go up there. But if you have space travel, if you have crowded people, if you have people with comorbidities, we need to start considering these things. So, yeah, and so do we have an example of why it matters to look up there to define public health down here? And we do. We work on it for a couple of years. It's a very curious case, as I said, and it relates to Calamniella pearsonii. So Calamniella pearsonii was firstly isolated by Venkat and his team. It was firstly characterized. It was one of the 20 full metagenome assembled genomes retrieved from the ISS environmental metagenomic sequences. And it looks like that under the electron microscope. So it was a novel genus that was firstly identified on the ISS using a metagenome to phenome approach. And it was named to honor Abdul Kalam, hence Calamniella. So it has a very close Indian link, who I'm sure you're familiar with, so I have to explain more. So, but then beyond MAGs, single-cell assembled genomes also retrieved the sequences of this novel genus and validated its presence in ISS samples. And then we had cultures, we isolated it. Metagenome, single-cell genomic approaches, they all agreed of its abundance. Just to say, it belongs to the gram-negative order of Enterobacterales. So it's not a spore former. And Enterobacterales are usually associated with things like Klebsiella pneumoniae, very well-characterized human pathogens. Initial bioinformatic prediction, when the genus was described, suggested that it has no human association and no pathogenicity. So that's the backstory. So what happened then is something completely random. A couple of years ago, we identified randomly Calamniella pearsonii in the saliva in Ireland, in our hospital, in the saliva of a patient with treatment-resistant schizophrenia. This doesn't matter, I'm just having it there for context. But it's part of another study we're doing to understand why people with schizophrenia are more prone to get infections. But we found it on the saliva of that patient. To say this patient was not infected by Calamniella was just present in the saliva. So we notified our clinical team, and what happened then is a couple of weeks later, we're notified of a clinical case in our hospital, CLABSI, which is a central line-associated bloodstream infection, that was associated with Calamniella pearsonii. And then a couple of weeks later, a second infection, and now we have 5 infections. Those are all human infections in Galway University Hospital, which we're affiliated with. We really don't know how many infections are actually out there. We have a brief idea through colleagues in other parts of the world, but we really don't know the prevalence of the pathogens. And it's very likely to be underreported because hospitals heavily rely on MALDI-TOF systems for identification, and in most cases, this is not part of their database, so they won't pick it up. It will come up as something else, so it will be misidentified. In Ireland, we just have an open hospital MALDI system. That's why we pick it up. So then we reached out to our colleagues to JGI, and we scanned MAGs, all metagenomes available to NCBI, to IMGM, and then we reached out to colleagues as well around the world, to clinicians. So practically, that's everything we have for Calamniella. We have 6 isolates from terrestrial ecosystems that they're non-human associated, 13 human infections, 5 of which are in Ireland, and some in the US as well, actually a couple in India. 9 MAGs from terrestrial ecosystems, and these 9 MAGs come all from the MetaSub project, come all from New York subway. So Calamniella on Earth has only been found in 6 isolates, 13 human infections, and 9 MAGs from New York subway. Nowhere else. We haven't picked it up anywhere else. From ISS, we have 44 isolates spanning 6 to 7 years of surveillance and 10 MAGs. So now we started scratching our heads because this is very strange. We have an Enterobacterales, which it's more abundant on the space station than on Earth from what we know. We know that's not the case. We know it's out there, but our genomic methods are not able to capture it as of yet. So So then, because we start collecting all the infections, we now have all the isolates in our collection, we started building a pathogenicity profile, and the pathogenicity profile started revealing commonalities. 9 cases of bacteremia, 2 cases of neonatal meningitis, 1 case of kidney stone disease, and 1 case of pneumonia. But most of the infections relate to bacteremia, so bloodstream infections. So I added that slide just to clarify something. How do we know Calamniella is actually the cause of these infections? Most of the infections are called CLABSI, right? So there's very strict, it's the same way we know any pathogen is causing any infection. There's very strict clinical criteria. So I just want very briefly to go through that. So we're on the same page again. So CLABSIs relate to central lines. Central lines are tubes like that they're inserted on a central vein in a patient to deliver therapeutics like chemotherapy, for example, or take blood and so on. So an IV line is like that. When you have an infection on the IV line, we score it from a score 0 to 3, which is beyond the point. But it happens in hospitals that you get infections on central lines. They're highly problematic because of the direct link of the central line to the vein. Therefore, the pathogen can directly enter the bloodstream, and once in the bloodstream, it can cause severe infections. So how do we know this is causing the infection? Well, the clinical criteria are the following. We have to isolate the pathogen on a pure culture from the tube. And then we have to also isolate the pathogen from the patient's blood in pure culture in 3 times from different sites taken 30 minutes apart prior to antibiotic treatment. So this defines that the infection is CLABSI, okay? And not only that, but the blood cultures have to turn positive at least 2 hours later than the isolation we have from the tube to know that it's the tube that is causing the infection. This is called differential time to positivity. So those are the clinical criteria, just to understand. So then, since we had all these isolates from space station, clinical, environmental, we decided to build an antimicrobial resistance profile on Calamniella. And again, we got a surprise because we identified a profile that's highly resistant to penicillins, and some of them that they're widely used in hospitals, like ampicillin, ampicillin sulbactam, but also to first, second, third generation cephalosporins, some fourth generation cephalosporins. And then towards the end, flight 10, which is very recent from the ISS, we started noting a non-susceptibility and resistance to carbapenems, which is last resort antibiotics. So then again, we're scratching our head. So we had 3 questions, okay, at this point. What is the gene that is causing this antimicrobial resistance profile? Because it's intrinsic, it's in all Calamniellas, it's not acquired. What is causing the bloodstream infections? And why is Calamniella so prevalent in spaceflight? Because other Enterobacterales aren't. So why? Those 3 questions we're still trying to answer. But we have some hints. First of all, to say that Calamniella is closely associated with Pantoea. So this is a gene network diagram. And you can see that the closest known species to Calamniella is Pantoea septica. Pantoea septica is also known to cause human infections, but it does not have this type of pathogenicity profile, and it's not very common human pathogen from what we know to date so far. So then we defined the core genome of Calamniella, and we looked and trying to answer these questions, and we pinpointed a couple of genes of interest. So we defined within the core genome a putative novel class A beta lactamase, which we're working now to characterize further, and we have preliminary called that BLA-MRL1. And this was present in all Calamniella pearsoniae we've sequenced to date, and highly conserved. And again, this is in par with other Enterobacterales. We know in Klebsiella pneumoniae, they also have a class A beta lactamase. Other pathogens like Acinetobacters also have beta lactamases in the core genome. So it's not unusual, but it's different. So we have to define this class A beta lactamase, and that would explain the resistance profile. The pathogenicity now, it's a bit more worrisome, because what we identified was that all Calamniellas were holding an aerobactin hypervirulence locus. Aerobactin, in plain words, 5 genes, and it's associated It's a siderophore, so it allows bacteria to survive in low iron environment, like human blood. And it's very well characterized as allowing Klebsiella pneumoniae to cause bloodstream infections, to the point where in Klebsiella pneumoniae, we have split all the genomes between classic Klebsiella pneumoniae and hypervirulent Klebsiella pneumoniae based on the presence of siderophores like Arabactin. But in Klebsiella, which is one of the most problematic human pathogens we have on Earth, only 5 to 15% of isolates have a hypervirulence pathotype. In Calamniella, it's in the core genome. Every isolate to date has it. So it's, again, strains. And the presence of Arabactin in animal models and human infections has been associated with up to 60% increase in mortality rates. Though, to say that, we don't have any mortality from Calamniella known to date. So then we tried to look a bit more, and we built a core genome phylogeny, including all Calamniella piersonii genomes and MAGs, and compare them to Pantoia septica, which I've mentioned is the closest associated species, plus some other yet uncharacterized novel Pantoia species that they seem to sit between septica and Calamniella. And so with yellow here, we have highlighted the ones that are from ISS origin. So you can see that the ISS origin ones are practically monophyletic. And but regardless of that, all Calamniellas have two things: Arabactin in yellow and the novel class A beta-lactamase. Okay. So it's which Pantoia septica and associated species don't have. So then we wanted to dig in a bit more, and we wanted to know where is this Arabactin pathogenicity island sitting. So we closed a lot of Calamniella genomes, and we found that they all have a non-mobilizable plasmid, which is this one here. All Calamniellas have it. And this plasmid has the Arabactin pathogenicity island plus a very cool feature, which is a virulence factor regulator, which allows for the pathogenicity island to be switched on or off depending on where the pathogen is. So if it's in the blood, it will be switched on. If it's on an environmental surface, it will be switched off to conserve energy. So those are the plasmids from 3 different Calamniella piersonii, one from saliva in Ireland, bloodstream infection in the US, surface of the ISS. And this shows how conserved this plasmid is. With red is the Arabactin, with blue is the virulence factor regulator. On top here, what I have is, we did find a similar plasmid in one Pantoia septica from a bloodstream infection, which was highly different, but it still had Arabactin and the virulence factor regulator. No, yeah, it does. I can speak through here, it's fine. Okay. So, yeah, so this is a phylogeny we built based on the class A beta-lactamase, but we're working to characterize it a bit more. The closest relative seems to be in Pantoia septica and Pantoia latae, but they're still only 84% on an amino acid level identical. So again, this signifies a distinct function. Just to clarify, Pantoia septica does not have the same drug resistance profile we see. So whatever happened to Calamniella, whatever this intrinsic beta-lactamase is, confers different resistance types. So then we went to the third question. The third question was, why is it able to survive on ISS so well? So what we did is we did NMR-based non-targeted metabolomics. And here we have the 2 Calamniellas that were isolated from ISS 6 years apart. So they were the 2 most distant we had in time points. And now we're working to characterize the intermediate ones as well. And 2 other isolates, which is a whole different story, Arthrobacter russicus, that's a fascinating other story I'm working on with Venkat and other colleagues, which is those 2 isolates are found. One of them was from Mir space station, one of them is from ISS, and this species has never been found isolated from Earth. So it has been found in 2 space stations and not on Earth to date. But again, that's a different story. So we defined Calamniella as intracellular metabolites, and then there were some commonalities. We're still working through the data, the very new data, but both isolates saw elevated polar metabolites like glycine, lysine, alanine, putatively in relation to support protein synthesis, stress responses, and so on. The earlier isolates showed high levels of formate and AXPs. The later isolate, which was isolated 6 years later from the ISS, had low lactate, elevated ATP and ADP compared to the earlier one, indicating maybe a reduced reliance on fermentative pathways and more efficient oxidative energy metabolism. So in other words, what we're trying to say is, has Calamniella changed metabolic profile over its presence on ISS, and how so? So in terms of its secretum, both isolates do not secrete aspartate or ornithine, which means they use it intracellularly. They release arginine, but F11, the later isolate, was also found to excrete mannose, cytosine, fumarate, extracellular sugars, again suggesting distinct carbohydrate and nucleotide turnovers. So the overall picture from the metabolomics to date is that Calamniella on ISS seems to prioritize intracellular retention of key metabolites, optimizes energy metabolism, and enhances stress defense mechanisms. They're all in par with its ability to survive in an oligotrophic, high-stress environment like the International Space Station. So, as I've mentioned, to sum up towards the end, we were not happy that we cannot find Calamniella anywhere else on Earth. So what we did is we developed a molecular assay, which is now validated, and it targets one gene that is highly conserved. We identified it to be highly conserved amongst all Calamniella genomes, and that's malate dehydrogenase. So our assay can detect 3 copies per reaction. The limit of detection is 3 copies per reaction with 95% confidence interval. So now we have a molecular assay. We can start scanning surfaces, particularly hospitals, environmental surfaces, even maybe the ISS, to be able to define the presence of Calamniella pearsonii on Earth and beyond. So yeah, we tested 54 isolates to validate our assay, 44 from ISS, 10 from clinics, and our exclusivity panel included Pantoea septica, several genomes, and other very closely related Pantoeas, as well as more distant bacteria. So, what are the unknowns? The unknowns are, it's Enterobacterialis. So, thinking of that, is it part of the human gut microbiome or any other gut microbiome? That's the first question. Second question is, what is the actual human infection prevalence? How many infections we have from this thing here on Earth? Okay? And third is, still the question to be answered, we have some hints now at least, why is it so prevalent in spaceflight conditions? That's why I'm saying that, so Calamniella, we would not know of this pathogen unless Venkat had identified it on the ISS. So this is very unusual. And again, it shows why we need to look up there to define public health here. But also, this, for example, seems to be much more troublesome for spaceflight than any of the human pathogens, bacterial pathogens we know on Earth. So that's what I'm saying. Infectious diseases in space are different. The troublesome pathogens will be different. So, and that brings me back to kind of a full circle to finish the talk. Not related to Calamniella, I'm not suggesting this happened, but it's this theoretical concept of invasive species reintroduction. When things go up there, and particularly when we have more people, more bioreactors going up there, more unregulated than the current situation is, our microbiomes will go up there, pathogens will go up there. Some of them, like Calamniella, they would have a chance to survive and propagate. They would change. And if they change and they accidentally transported back to Earth, they could be different enough, and that's completely theoretical, but they could be different enough that they can be established and spread upon susceptible native hosts, plants, humans, microbiomes, and animals. So, going back to One Health, maybe the current term of One Health that includes planetary health and includes summarizing everything, all the activities that happen on planet Earth, maybe it starts not being sufficient enough to describe our human activities. Maybe we now proactively, like in the case of Calamiella, need to take approach to health in space that can ensure the safety of public, celestial health, but also the health of space, of astronauts or people who are in space habitats. So we do envision a future where the health of all terrestrial and extraterrestrial environments is considered, and embracing a new era of health collaboration that goes beyond our planet, but to a truly multiplanetary scenario. And with that, just want to thank key collaborators on this project and funders from Ireland. And thank you all for listening. I'm happy to take questions.
That's truly a very big picture beyond my imagination. So there are a lot of questions there. I would love to start from
here. Hi, over here. I was wondering, do you have some sort of controls to be able to understand whether this Calamiella that's on the ISS has evolved on the ISS? Is it known to be growing there? Is it known that this is the same progenitor strain that was there 6 years ago that's there now, and so on and so forth? So for example, if you look at any other bacterium here on the Earth, would you see similar kinds of changes, right? Two different kinds of pathways and so on that you're seeing from metabolite profiles?
Yeah, that's a good question. So for Calamiella, we're lucky enough to have a lot of isolates from the ISS, which is really cool and rarely happens. We have 44 isolates spanning 6 years. So we can do, we have looked, I saw the slide with core genome phylogeny, and they seem to be highly conserved. So meaning that the genomic differences are very subtle, even from isolated 6 years apart. But as Venkat was explaining earlier, what happens during spaceflight is usually the differences you see are on metabolomics or transcriptomics more so than genomics. So that's why we look more on the metabolomics. So now we have characterized only the two most distal isolates, but by getting a picture of the metabolomic adaptation over time, which again, we're lucky enough to have all these isolates.
Sorry, just to interrupt. I guess what I'm trying to say is because reintroduction is also presumably happening with all the people that keep going up, how does one control for that as a reintroduction versus in situ evolution that's happening on the ISS?
Yes, that's a good question. So from the analysis we have to date, from all the Calamiellas we have, the ones on ISS seem to be monophyletic. So they seem to be their own distal group. They're deviant from the main Calamiella lineage, and they're completely distinct from the ones we see on Earth. So, but again, that's something that commonly happens. You may have one isolate that goes up there through cargo or crew, and then for one reason or another is able to survive. And if it survives due to the environment being oligotrophic, you don't have a lot of competition, is able to establish itself, and if it's able to persist over years, like in this case, then you have.
The hypervirulent strain of Calamiella, did you also find them to make more mucus? Like some of the Klebsiella strains are hypermucoviscous, and do they also have a potential to form more biofilms, stronger biofilms?
That's both very good questions. So so the answer is we have checked for both. In Calamiella, we have now distanced ourselves in clinics from hypermucoid phenotypes and hypermucoviscosity for defining hypervirulence, because we found it that is non-specific. So that's why we look at the genomic markers instead. But to answer your question, for Calamiella, some of them are hypermucoid, but some of them aren't. So we don't have, this is non-specific to describe the presence of Aerobactin. The second question related to biofilms, So that's a very good question. So when a group in India, actually, when they first reported a clinical case of Calamiella, they defined it as biofilm former. This is something that in our experiments we haven't been able to replicate. But given its ability to form, to cause clubsies, because catheter central line associated infections usually are by biofilm formers, it might be the case. But the isolates we have do not seem to possess this phenotypic ability.
Yeah, a very quick question. In the interest of time, I would like to ask you to catch hold of Georges in the lunch on Earth with food. So please feel free to hold. So
I'll just make an announcement. Thank you, Georges, again for a great talk. So I'll just make an announcement and then we can sort of hand over mementos. I know I'm holding you guys from lunch, but we have an excursion planned for this afternoon. So just a couple of announcements about that. The session for today afternoon will start at 2 p.m. instead of 1:30 p.m. We've sort of pushed it back by half an hour. So the reason for this is that this gives you, if some of you want to drop your bags off in the hotel or in the guest house before you leave for the excursion, then there are buggies available right now. So feel free to drop that. You don't have to. You're welcome to take your bags with you. You can leave them there. You can leave them here as well if you want, whatever you please. But if you want to go back to the hotel, this gives you time. So we will leave. There are buses that are going to come here right outside the building at 3:30 p.m. So that's when we will leave. And it will be about somewhere between 9 and 10 is when we will return tonight. You can take this.
It's here. Yeah.
Can I steal a bottle of water? No, it's all right. Thank you. Thank you. So good.
Okay, welcome back to session 6. This is on computational approaches for microbiome science. This session will be chaired by Dr. Virendra and Professor Mayappan. I'll introduce them. Dr. Virendra is a research scientist at IBSE over here. He completed his postdoctoral research at the University of Vienna and University of Copenhagen. His research interests lie in bioinformatics and data science with a focus on transcriptomics, non-coding RNAs, and healthcare applications. And Professor Mayappan is an assistant professor here at the Department of Biotechnology in IITM. He earned his PhD in chemical and biomolecular engineering from NUS, Singapore. His research interests include systematic bioprocess development for biopharmaceutical and cultivated meat manufacturing, data-driven cell line development, and other computational system biology approaches for clinical marker identification.
Thank you, Mazia. Welcome back, everyone. And I have the honor to introduce Professor Karthik. So everybody, of course, knows Professor Karthik. So he's professor in the Department of Data Science and AI at the Vadhavani School of Data Science and AI, IIT Madras. His research group focuses on developing scalable algorithms and computational tools to analyze, predict, and manipulate complex biological networks. His work spans synthetic and systems biology, and key areas include microbiome systems biology, in silico metabolic engineering, biological networks design, and big data analysis in biology. He also coordinates the Center of Integrative Biology and Systems Medicine at IIT Madras. So please, everybody sit back and enjoy the talk.
Thanks. I'll not thank myself, but I'd like to thank Karthik and Venkat for really, mooting the idea of this conference again and going through with it. So very happy to be speaking in front of you. And I also seem to be really outnumbered here in this conference. We have as many Karthiks in the conference as we have computational people, it seems. So, so I think, this will be a good place to start looking at computational approaches to one sec. Just a moment. Computational approaches to study these microbiome data sets. Okay, that looks perfect. I'm going to skip this slide because this is exactly the intro that Virendra just read out. But just to say that we focus a lot on microbiome systems biology, also on some biological data analysis, multi-omic data integration, and so on, with the goal of impact in the clinic and the environment. These are some of the interesting algorithms and applications that we've developed over the years. So I'm going to focus on maybe a little bit on some of the things in blue. I'm not going to talk much about the other things. And you will see that there is one poster here and also a talk tomorrow by Virendra. And it will be great if you can interact with my student at the poster and also Dr. Virendra tomorrow. I don't need to really introduce the microbiome to this audience, but one thing I want to underline and maybe argue at Dakshin Chitra is what is the importance of metabolic interactions? How important are the metabolic interactions? Because they seem to predominate in a lot of these communities, and in certain communities we've been able to understand them well, in certain other communities we haven't been able to yet understand them. So how important are these metabolic interactions? Because we are biased towards the metabolic lens of viewing at a cell. And we've been trying to understand cells through the metabolic lens for the last several years. And we are looking at this both from the perspective of building, say, microbial consortia for metabolic engineering applications and for studying various extreme environments, right from deep sea to outer space. Believe that understanding these interactions is crucial, whether it be for whatever the application be. Right? And how can we understand these microbial interactions in communities? And the question is, can we use metabolic models? Many of you may have seen such nice diagrams, especially if you have a colleague biochemist, you would have seen this on their wall. So essentially a representation of all the metabolic reactions happening within a cell. These kinds of models have been reconstructed for lots of organisms, and you have some fantastic groups, including from University of Galway, right, where they've been building a lot of these models in Innes's group and so on. And these have been constructed for a lot of organisms, essentially trying to capture what are all the key reactions happening within a cell. And on this basis, can we start predicting how the cells will interact, talk to each other, and so on. And some of you may have seen these wonderful databases as well. And there are many methods to study these networks. But ultimately, we try to simplify this, right? So this is how I encourage my students who are not very adept in biology to really think of the cell as just a bunch of nodes and edges or a bunch of mathematical equations that capture the complexity of metabolism. And how do we model microbial communities? There are many methods that exist, but I'm going to focus on a couple of families of these methods today. So constraint-based methods, which are very useful to study fluxes and whether a metabolite is being produced and so on. And graph-based methods, which do not capture enough of the dynamics, so to say, but still give you a very interesting perspective of what kind of things are possible within a cell. And there are, of course, many more methods that are being developed, and the applications are numerous, as I will demonstrate to you. So in constraint-based modeling, this is very popular, especially in the space of, this was literally started off by chemical engineers like Bernard Paulson. And it's very popular for applications such as metabolic engineering, and is able to predict growth rates or reaction fluxes based on certain assumptions and so on. But it demands very well-curated models where you have an accurate understanding of system stoichiometry and things like that. On the other hand, you have network-based models where you can do things like pathfinding on networks and so on, essentially sort of the Google Maps within a cell, and metabolic exchanges and predicting new pathways that are, if they can be synthesized, new drugs or new metabolites can be synthesized by a cell and so on. Some of these require atom-atom mapping and things like that, but others, like I will show you, can just work in the absence of much information, just working with structural information and reaction lists. In any case, we would require methods that are very scalable, that are accurate, and can handle, say, things like uncertainty in various ways. I'll get to that in a moment. So this is a broad overview of some of the algorithms I'm going to talk about you. So these two are constraint-based approaches. These two are graph-based approaches. I'm going to keep it fairly high level except for the first one. In fact, second one is just this slide. But I'm going to do a little bit of a deeper dive into the first one because I think it's very interesting. Even at the risk of amplicon sequencing getting or sounding quite dated. So this is our new approach called pan-genus metabolic model. So some of you may be familiar with this GSMM acronym, which is for genome scale metabolic model. So here we are proposing that we can build a pan-genus metabolic model to capture a single genus, right? So let's say you want to capture Bifidobacter in a particular system. Can we use these PGMMs to capture these this entire genus? Right? And this was developed by Indu, a really bright student. She was a PMRF scholar. She's close to finishing her PhD and is in the US right now. And she was also co-guided by Professor Himanshu. So the motivation is just this, that there is a ton of 16S data out there, historic data, currently data that people are currently gathering, and there are not enough tools to leverage this, right? So 16S data is obviously low resolution, so to speak, in that you may not find out the species, but you'll have only resolution until say the genus level. But if you want to actually build one of these metabolic interaction models that people have been building over the last several years, you need species level resolution. So how do you sort of tackle both these challenges simultaneously? Right? And I've not gone into this, but this is stuff I usually teach, usually lecture a lot about. Constraint-based models are super useful for studying microbial communities. There's an array of methods that people have developed, like SteadyCom, OptCom, MyCom, and so on. You see the COM that stands for community in all of these cases. But they're typically specific to microbial strains or species built from whole genome sequences. You could use MAGs as well, but typically proper high-quality whole genome sequences, and a lot of manual curation goes into it after that. But then a lot of data that's around us is 16S, and which cannot give you equally good resolution. And we believe that no existing tool has effectively addressed these complexities of inferring something meaningful in terms of the metabolic spaces, metabolic interactions, using 16S data. So how do we address this challenge? So our question is, can we build genus-specific metabolic models and yet, to some extent, whatever extent possible, mitigate the uncertainty in inferring metabolic capabilities? Because the goal of any genome scale modeling is say, hey, how much of X is going to be produced by this organism or this community or whatever. So can we do this to some extent with some caveats in the case of composition uncertain models like you will be able to build when you have 16S data. So this is the algorithm, we call it Panera, the framework. So we construct pan-genus metabolic models using genome scale metabolic models from what is known as the Agora database. And those of you who closely follow the field may have seen another very nice paper in Cell Systems last week called Apollo, which has like close to 250,000 models that have been built, right? So most of these are draft models, but many of these are well re-annotated and so on. So using these kinds of models, I'll tell you why we use from a particular database. We develop species-aware pan-genus models that can help you understand the metabolic capabilities of a desired genus and trying to include whatever level of resolution you have at hand, right? So if you know the species, go plug in the species. If you know only the genus, just put in the genus. If you know species probabilities, I know there is X of, there is 10% of Bifidobacterium adolescentis and 20% of Bifidobacterium longum, go and put in that data. So we allow this kind of different resolution data to be merged together. And we use 3 simple steps. First, we build a combined model, which is just a union of all the reactions and so on. But they are not just pooled like some of the other people do. We also have a special biomass objective that tries to represent all of these. And we add parameters to accommodate the variation in species composition. So we essentially do something like this. So the pan biomass is constructed from the individual biomasses into the probability of each of the species probability that is known for, or abundances that is known for each of the organisms. And we use a reaction species vector and a species probability vector and all that. I'll not get into all the details. But what you have is this. You have multiple species, some known, unknown probabilities. We merge this to build a single model at the end of the day, which we want to use for various predictions. Okay. So these are our questions, right? This is what I want you to take home. So can PGMMs explore the metabolic activities of a genus, right? And this answer to the second question seems yes. Like, so does metabolic potential vary with species proportions in the genus? But there is an interesting caveat there. So even if you have uncertainty in certain organisms, it looks like certain metabolites across a particular genus are similarly produced. So we can probably tap into that. And can we employ further review if we have ambiguous or unambiguous taxonomic data to study and predict personalized microbial communities? Right? So these are the questions that we set out to answer. And what we believe is this PGMM is a useful tool. I'll just get to this in a moment. So what we start off with the species probability vector and then customize the PGMMs based on these species probabilities. We do what's known as a flux variability analysis, which tells you what's the least possible of some X metabolite that can be produced and the max possible of that metabolite that can be produced under a given set of conditions. And we look at this band, and this is what is interesting, right? So some things may be tightly regulated, there's like not much variation. Some things might be all over the space, and something may be having low variation. So what is interesting is that, for example, this is very interesting because it says that despite the uncertainty in composition, I'm very confident about how much of this metabolite is going to be produced. It may not be for a large number of metabolites, but if this is happening for certain SCFAs and short chain fatty acids and so on, that's all you might want to know about, like say a gut microbiome model to assess the health of a gut and things like that. Right? And we evaluate this flux range and we classify the bandwidth type based on various calculations. None of them are too involved, but it's fairly straightforward. And these are some of the real model simulations that I'm showing you here. So if you see here, there is acetate, cysteine, and so on. But if you see acetate is in bacteroids is very, very tightly regulated. Whereas it's all over the place in Streptococcus and in Prevotella. Right? And this is on different diets, so you can start exploring. There are ways to capture how diet influences the microbiome or like basically the availability of nutrients and so on, and use that essentially to figure out what is the variation. Right? And we see that this is very tightly regulated and this is low variation, and this is extremely high variation. Right? And what you see on the right is that we've clustered some of these species based on their metabolic similarity, reaction similarity, or flux bandwidth. So this tells us, see, this is a question that all of us typically have when we look at these, especially gut microbiome data and so on. You see two very different gut microbiomes that from two equally healthy people or so on. So how do you rationalize this extreme variation that you have in these gut microbiomes? Are they still having similar metabolic properties? What are the real markers of health and so on? I'm not saying this answers the question, but these are some things we really wanted to think about, and this gives us a lot to think about. So this helps us understand what is the metabolite production bandwidth of a genus, and how in a particular context the similarities between different genera will change. So this is a very interesting way of looking at it, I thought. And we also are able to build hybrid models. I'll not go into all the details. So we compared with 3 different kinds of models. One is a PAN model, which is already available in things like the Cobra toolbox, for those who know of the toolbox. It's literally like a bag of enzymes model. You just put all the organisms into one bag of enzymes and see what can be produced and so on. And what we compared against was just we don't have hard ground truths except for the last thing, which is about which is from metabolomics data that we have from publications. We compare with the genome scale metabolic models that are available and using that, right? So that is high resolution. And you drop that genome scale metabolic model and just use the genera and the genus information to build these PGMMs and simulate. So what we found that we are able to have at least a 10% reduction in the prediction error when we integrate, we use our modeling approach, especially with the varied resolutions, right? So where I know species, I'm able to plug in species. Where I know genus, I'm able to plug in the genus and so on. Anyway, so the bottom line is, this is just a new way to try and see that with limited data like 16S, can we still make certain meaningful predictions? There are obviously limitations to this approach, right? First thing is the quality of the models that you go in, and this is true to any modeling approach. Although resources like Agora are being like updated continuously, and just Apollo came out like last week, where they have 19 phyla and about 247K new models. The catch is that we need consistent annotations to be able to merge and have these models talk to each other. Otherwise, it becomes a nightmare. This is something that's been plaguing the field from, say, the last 2 decades, and we've already written a paper about it in 2015 about the challenges in reconstructing and merging different kinds of models and so on. So this is one big challenge. But other than that, this of course, this modeling technique inherits all the limitations of constraint-based modeling approaches. But that apart, I think it's still a very potent method to interrogate these kinds of systems. So to quickly summarize this part of the work, there are not enough methods that can exploit 16S data for modeling. And most models, current methods just lump them into like PAN models and so on. What we create are adaptable species-aware pan-genus models that can minimize uncertainties in various ways while making predictions and so on, and ultimately try to tell us what is a genus's contribution to a metabolic community in a particular environment. And our hybrid models outperform the conventional GSMMs in predicting the metabolic capabilities of a community, even with uncertain taxonomic information. So that is like the one major study I want to talk about. I'm not going to go into the details of this. This is the only slide on this method, but the idea was to build function-specific minimal microbiomes. So if you have a particular environment and you have a particular objective, let's say, I want to produce X amount of short-chain fatty acids and so on, we are able to say what should be the minimum microbiome that's available there to produce this, right? So this helps us to study systems in various ways. For those who are interested, can go and read about the paper, but especially those who are working with gut microbiome data and so on. With that, I switched to, these were constraint-based approaches. I tried to put one small icon at the bottom. And now I'm going to switch over to graph-based approaches to studying microbial communities, where the idea is far simpler. There are various ways to represent metabolic networks as graphs. And so if you think of Google Maps, every junction is like a node and every road is an edge in the network. Right? So how do we represent nodes and edges in this network? There are other approaches which I have grayed out because they are not very useful. The one major way to represent them is something known as a bipartite graph, where you have 2 kinds of nodes, so circles and squares. So the circles represent metabolites and the squares represent reactions. And what you have are links between circles and squares and circles. No links between, so basically ATP and glucose connect to R1 and outcome glucose 6-phosphate, and ADP. Right? The problem with this previous kind of approach is you might convert glucose to ADP to pyruvate and have a two-step glycolysis. Right? That's obviously not correct. Whereas here you know that you need both these metabolites to produce the metabolites here and so on. Right? So you can be very correct in terms of the metabolism in the metabolic biochemistry when you use this kind of a model. And this is how we built. So now we use this to actually represent the community itself. So you have B from metabolite B from organism 1 that comes into an external pool, maybe if it's secreted, and then it is taken up by organism 2, and that becomes B2, and this is able to affect the metabolism of the second organism. So this is our central hypothesis. There are some microbes exchanging certain metabolites, and based on this, their metabolism changes. How does this change? This is the question that we wanted to ask. So quickly, what are the key features here? MetQuest is our algorithm, which requires only the topology of the reaction network, can scale well to large metabolic networks. We've tried it on large communities and so on. And it can efficiently handle cyclic and branched pathways, which is usually a problem when you work in graph theory. And it also is able to capture various alternate routes of conversion and so on. So with that, now that we have this methodology to identify pathways and so on, can we use it to quantify the metabolic interactions? So there we built a simple index called metabolic support index. The logic is basically this. How does one organism help in improving the metabolism of another organism in the community? So you put two organisms together. How do they interact? Does one help activate certain reactions in the organism, or does it make no contribution, or this is like a sort of parasite that grows on the other organism by taking a lot of its metabolites and so on? We don't look at the rates of these things which are typically necessary to find out if one is exploiting the other, but we can say whether A benefits more from the interaction with B or B benefits more. So this is obviously a directional relationship. So in this diagram you see that A gets a benefit of say some 30% from B, whereas B gets a benefit of just 1% from A. So this helps us, this is one way to predict, and we've done some experiments and looked at, one rare time we actually did experiments, and we've also looked at studies in literature which have shown in many examples that the organism with higher MSI grows much more in a co-culture and so on. So if all the reactions in the organism remain stuck, there is no benefit, but otherwise, you have one organism benefiting the other in different ways. Right? As I just said, the organisms with higher MSA seem to grow better in a community. This is another important slide, maybe the only last important slide in my talk, where our graph models help address many questions. So this includes what are the metabolic capabilities of an individual organism or a community, and these could also be addressed using constraint-based models. What are the metabolites that are exchanged between organisms? You'll see them, we've explored this in some of these papers. And how do organisms depend on one another? Right? What kind of dependencies are there? And how does a community evolve? Right? So are there unique contributors in a community? What are the keystone species in a community? And and the next thing we are really interested in exploring is what are the trajectories possible for these communities. So that's something that we haven't made enough headway into, and it's something we are excitedly working on. And now I'll switch to certain applications. I think I have a few more minutes. So the first is something work that we did with Niranjan. I hope he might talk a little bit about it tomorrow, so I'm not going to dwell on it. But the idea was that how does this cartoon was made by Niranjan and his team. So wherein this likens the gut administration of antibiotics to the effect on the gut to a forest fire. Basically, the whole gut is nuked, and then slowly it blooms back to some sort of normalcy. Right? So how does this happen? What are the microbes that help here? And are there? It turns out that certain folks can recover faster. And can you predict that using the metabolic interactions and so on? So this is something we delved into. A very interesting piece of work. Thanks to Niranjan for initiating this fantastic analysis. And this is something that Venkat was alluding to in his presentation earlier. So we wanted to understand the key metabolic interactions in the ISS microbiome and focused around Klebsiella. So what we did was we looked only at locations where there was a significant presence of Klebsiella, and we did a bunch of metabolic modeling. So this is the graph-based analysis, and this is the constraint-based analysis. We did a bunch of these analyses to ultimately try and predict these kinds of things, right? So what are the microbes that are positively affected by Klebsiella? What are the microbes that are negatively affected by Klebsiella, and so on. And here we found a particular interaction that Venkat was able to confirm in the lab. So we found that under microgravity conditions, Klebsiella has an even stronger effect on Aspergillus. So you see that the structure is completely destroyed in microgravity conditions, whereas it's quite healthy in normal gravel conditions alone. And so it's very interesting to see to throw out these kinds of predictions and see how we are able to explain some of these in the lab setting and so on. So we did a bunch of studies. I'll just talk about a few points here. So our pairwise interactions and community interactions were able to suggest various roles for the different organisms in these communities. Basically, these kinds of modeling approaches try to help unravel what are the key players, what might be the key players, and how the interactions that they orchestrate are able to get the community surviving and proliferating and so on. And we found how Klebsiella and Enterobacteriaceae interact with each other as well. So last couple of minutes, I'll just go into this other quick study, which was led by Pratyay and Venkat. So Pratyay is in the audience if somebody wants to talk to him. So here we focused on Enterobacter bugandensis. And we did a lot of analysis of, here we also did some bioinformatics analysis trying to understand what are the differences between the terrestrial strains and the ISS strains. And we found some really interesting prevalence of MDR genes in the ISS strains and so on. And we basically again try to understand how Enterobacter regulates the metabolism of other co-inhabiting bacteria. And this was something very interesting for us to look at in this. So with that, I think I'll just quickly conclude. I think I mentioned most of these. We found also some reciprocal influences between Enterobacter bugandensis and Klebsiella. So it is quite fascinating for us to look at all these complexities in these metabolic models and their interactions. And this was a study that I did with Karthik. So this was my MS student who's now doing his PhD with Karthik, where we looked at deep sea hydrothermal vents and what kinds of really interesting metabolic dependencies are there and so on. So this is just like a high-level overview of how we can apply metabolic modeling to study various interesting systems. With that, let me acknowledge all my collaborators. I think a fantastic array of collaborators over the years, many of them in the audience just by chance. And I'm very happy to take any questions. Thank you.
Thank you, Professor Karthik, for the fantastic talk and the wonderful tools to work with. So we have good amount of time. Yeah, questions, please. Oh, yeah.
So very good talk, Karthik. So we know the pangenome of the species and genus are not of the same size, right? Some have bigger pangenomes, some have smaller pangenomes. We also know, and this was just my recent work where we showed that not all the strains for a species, they occur at the same number. Some species may occur at more strains, some may occur at smaller strains. So how do we square it off using all the metabolic networks that you are making, including the pan-genus metabolic network and other ideas?
So we can embed all these probabilities, right? So depending upon the variation, right? So you may say that in certain guts, let's say in European guts, you had a slightly different proportion of these species. And in American guts, you had a different proportion of these species. So we could build different pan-genus models to compare these two even. Right? So based on whatever is your guess of the species abundances of the different species in a given genus, right? And then you can start putting these genera together in larger community models and predicting.
So you are saying that we can also explain this metabolically, that why certain species they need a bigger pangenome.
I'm not sure.
So the point is, let's say like for Bacteroides ovatus versus Bifidobacterium infantis. Bifidobacterium infantis, you have a very tiny pangenome. Bacteroides ovatus, you have a much bigger pangenome.
Yeah, but I suspect maybe you could just explain that with its broad metabolic capabilities without like resorting into too much modeling. Maybe they don't produce too many fancy metabolites, right? Whereas some have an array of metabolites that can be produced in an array of environments. right? So that could even explain pangenome size. I'm like not too sure about that. But here the main idea is that if you have different hypotheses about the structure of a genus, you can now plug that into this model and use this to maybe ask a question of which is the more meaningful composition.
Thank you, Professor Karthik. Yeah.
That's a wonderful talk, actually. So I wanted to ignore my ignorance if it is silly. So the thing is that I wanted to ask you, given that the metagenomics is producing a lot of genome and majority of the genes in the metagenome for which we really do not understand the function for, how does that impact this kind of modeling?
Absolutely. I think that's a great question. That's something we want to focus on. And this is a question I think I got last time as well from Venkat. And this dark matter is something that's really exciting. And hopefully, I don't think potentially one aspect we could look at is that when we have some metabolomic data, we can use it to retrofit these metabolic models. You see what is not being captured in these metabolic models, and we can start coming with hypotheses of how these reactions may be possible in this genus and things like that. We could use that, but as such, we are very much ignorant of the, or agnostic of the dark matter, which we haven't yet explained. So that is definitely a challenge. I also suspect that we could develop some methods to sort of pool these unknowns into some certain factors, but we also need good data on the other side, right? So I don't know what is the spectrum of metabolites we've been able to estimate too. So what part of the metabolites are unknown, right? So there is, it's exciting, more conferences.
Karthik, thank you so much. It's a wonderful talk again. Happy to be in this conference right now. So I learned a lot about, read a lot about metabolic models. My only question is that, is there a way to sort of steer it to a certain pH or steer it to a certain temperature, the kind of interactions which might happen? Is that, or can that be incorporated in this metabolic modeling?
Not directly, but see, the way these metabolic models work is that, in fact, there is also a lot of talk about how do you integrate expression data into these models, right? And there are lots of methods to do it. So for instance, if your temperature or pH or things like that change, or the environment changes, your gene expression is going to change. That is a way to plug these back. Essentially, my starting point is a long list of reactions. So that list of reactions changes based on the conditions. So if I have a way to map it, that should be doable, definitely. So like under certain temperatures, certain reactions will get deactivated, certain enzymes will get denatured, and certain reactions aren't going to happen, right? So I can plug that information back into that model. I don't think modeling can answer that question directly, but if you have knowledge on the effect of conditions on the model, you can directly incorporate it. It's very possible. Thanks.
Great. More questions? Yeah, one more.
Sorry, this is just so interesting. So on this, carrying forward the last question, so we know, let's say the vaginal microbiome is like extremely acidic, but if you have bacterial vaginosis, the pH increases, right? So, and we know the members in that case also are altered. So I'm just trying to take this leap of faith. How will we model this phenomena using a metabolic network? So for example, in a healthy vaginal microbiome, you have just 1 or 2 species, and there you have very low pH. But if you have vaginosis, you have other type of species, the diversity increases and the pH also increases. So if we have this toy experiment, how would we model it using this approach?
That's a very good question, and I think it's possible, right? I can already see a way to do it in terms of trying to sort of reverse engineering, right? So this is like also going back to that minimal microbiome study that I didn't talk about. So you say that, hey, what are the minimal set of microbiomes that are needed to produce this kind of acidosis? Right? And go back and see, and the thing is that I didn't talk about is we can also say what are the different sets of organisms that can do that. So you can now get a list of alternate things that you want to examine. So maybe you can, I don't think you can, this will be very good for hypothesis building, which we can then test out in a lab. Right? You can say, hey, it looks like maybe organisms A, B, and C are likely to be higher in the case of vaginosis, and you can use that to maybe go back and see if this is actually the case and so on. We could give different candidate combinations of microbes that could be responsible for these conditions potentially.
So to add something on that, Karthik, to answer his question, how do you model? Take that sample, do some mass spec, and get the metabolites, give it to him. So that would be the best answer for the subsequent analysis. You don't need to do every single time the run. So that's what we are trying to do: collect the samples, extract the metabolites, do the mass spec, and give the list. So he can try to model.
Great. I think. Okay, one more question back there.
Thanks, Karthik. Amazing talk as usual. We are a startup and we work on breast cancer therapeutics. So your gut genome is just a casual question. Will the gut genome or gut microbiome, will it change for any cancerous patients with a normal patient?
Yeah. See, I think there are lots of correlations, but I haven't yet seen causations. But there are like lots of strong correlations that people have seen. So it's not clear as to what drives what. I'm sure there are other people in the audience, including maybe Niranjan, who might answer some of these questions tomorrow or at another time. But lots of interesting things happening. And one thing we are trying to do in many of these cases is when we have two data sets, right? So diabetic, non-diabetic,
Exactly.
cancer, non-cancer, we are trying to integrate metabolic features to see if it aids prediction. And currently in a preterm birth cohort, we seem to get like a lot more accuracy when we are including the metabolic features in prediction. But that is still not to say any causation, but again, strong correlations possibly.
Once when you get the money, we'll come back to you. Thank you very much. Thanks
Great.
a lot.
On that note, let's again thank Professor Karthik.
It gives me immense pleasure to introduce the next speaker, Professor Narendra Dikshit. Professor Narendra is a professor of chemical engineering in IISc. He earned his PhD from University of Illinois. His research interests include computational biology, infectious disease, theoretical immunology, and microbiome studies. I guess he's going to talk about some microbial community modeling today. He's also recognized with several honors. Some of the notable ones are the Senior Fellowship from Wellcome Trust DBT India Alliance and the Indian National Science Academy's Medal for Young Scientists. So let's welcome him.
Am I audible now? Yeah. Great, thank you. Thanks again for those kind words. I'm going to tell you today a little bit about linking interspecies interactions to community structures. Karthik has, of course, done a wonderful job of setting the stage up for thinking about interactions. Before I start, let me acknowledge the people who have done all of this work. This work was done primarily by Amir, who was a graduate student in our group, and then Gayatri, who incidentally was a PhD student with Karthik and then spent time in our group as a postdoc. And then colleagues from Unilever who did several of the experiments that we have used to test some of the theories that we have developed. So at the outset, I thought I'd start out with this sentiment that movies are ahead of life. And I wasn't so sure of it until I came to this conference, because I realized that our movies, at least in the ones in Bombay, have anticipated all of the confusion that the two chairs of this conference are going to create. Right? And so they made this movie called right. So, I haven't seen this movie, but I thought now I should go back and look at it. And so let me take this opportunity to thank the two Kartiks for putting this wonderful meeting together and for having us all here. I've had a great time the last day and a half, and I'm looking forward to the rest of it. So back to serious matter. So the goal of what we have been trying to do here is to see if we can construct theories that help us predict structures of microbial communities. And by structures, we mean the identities and the compositions, relative compositions of the microbes that constitute a particular microbial niche. And I'm going to tell you first what the challenge is. And Karthik alluded to it. one of the challenges that at least that we've been looking at are the presence of these high order interactions, which render experimental investigations of these phenomena difficult. We've constructed an algorithm called EPICS, which I will talk about in some detail, that helps us address these high order interactions and still make some progress with trying to predict structures of communities. And then towards the end, I'll talk briefly about another method that we have constructed, called, which we call the quartet, which helps us disentangle cooperative and competitive components of the interactions between species. And throughout, I will draw links to genome scale metabolic models and also synthetic communities, which help us test some of the ideas that these algorithms present. So let me start with building up the case for high order interactions. This we came across because of some experiments that our colleagues at Unilever did. They studied a 7-member synthetic oral microbial community. They actually studied an 8-member one, but for this purpose, I'm going to talk about a 7-member community. In the later part, I'll talk about an 8-member community also. So these are apparently important for things that go on in our mouths. And the experiment that they did was put together equal amounts of each of these entities and then measured the abundances after some 72 hours. And many times they chose all of the species, sometimes a few of them, and so on. And what they found was rather interesting. They focused on one of these species called A. viscosus, which in monoculture had the highest abundance. Then when it was grown together with some of the other species as pairs, its abundance suddenly dropped. So in this case, for example, this A. viscosus has been paired with L. kz. And this is the abundance of A. viscosus by itself, and this is its abundance in the community of this pair. And these are the abundances of the complementary species L. kz. So nothing happens to L. kz, but A. viscosus comes down drastically. And that happens with another species of that community and a third one also. But then when all of the 7 species were put together, A. viscosus came up almost to the levels at which it was when it was by itself. So this was an intriguing observation that suggested that there was all this repression that was going on in pairs, but then a rescue that happened when many of these species were brought together. How could this be? So to understand this, we had to think about high order interactions. And what do we mean by high order interactions? Imagine a species 1, which is suppressed by some metabolite that is produced by species 2, and also independently suppressed by another metabolite perhaps that's produced by species 3. But when 2 and 3 are simultaneously present in this milieu that also has 1, let's say species 2 produces some other metabolite, 3 yet another one, and these metabolites react outside this extracellular medium, and then they perhaps have a beneficial effect on species 1. This now constitutes a high order interaction because you need the presence simultaneously of these species 1, 2, and 3 for this interaction to be realized. It's possible that these high order interactions might be involved in the rescue of A. viscosus that they saw in the previous experiments. We are not the first ones to see high order interactions. These have been seen earlier, and they've been argued to have a stabilizing effect, which was another reason for us to pursue this line of approach. And in this paper from Roy Kishoni's group, they set up mathematical models using these generalized Lotka-Volterra equations, which tell us how populations of individual species in a community evolve as a consequence of their own individual growth, but also supplemented by interactions from alternative species that are there. This is binary interactions, pairwise interactions, and here are terms that quantify higher order interactions. So this is a ternary interaction, which involves the simultaneous presence of species I, J, and K. And this term here, Bijk, represents the influence that species J and K together have on species I. This is a quaternary interaction, and so on. So what they found, this is just to tell you what these orders are of the interactions. What they found was the stability of communities improved as they introduced these high order interactions. So for example, if the community had second order interactions alone as the highest order of the interactions, then a large number of cultures that they simulated using these interactions went extinct. Whereas if they introduced fourth order interactions, then they found that those communities remained stable. So our question was, could these interactions then be part of what rescued A. viscosus in those experiments? So we said, let's try and quantify the interactions using the experiments that were available to us. And the way you quantify them is to look at the abundances of species in the monoculture and the abundances of the same species in higher order systems and look at the differences, which then tell you, give you a measure of these interactions. So for example, if I look at this culture of A. viscosus and L. kz, then depending on the differences in the abundances that I have here, I can identify what the magnitude must be of this interaction coefficient that explains this data. And if I do that, I find that LKZ and S mitis, which are these two species, they have the highest negative interaction on A. viscosus. They suppress the population of this A. viscosus the most. So now what it meant was that the other species that were there somehow had to overcome the negative effect of these two species and rescue A. viscosus in the all species community. You might ask which of those other species are involved and to what extent. So to answer this question, our colleagues at Unilever set up what would be called these full factorial experiments. So here, the species that are involved in a particular experiment are on the left y-axis. The experiments are numbered on the x-axis, and species that are filled are present in the experiment, and those that are absent are shown in open circles. These experiments all had A. viscosus, LKZ, and S. mitis, because these two species were going to suppress this. And then you either introduced one or more of these other species, trying to find out the combination of those other species that would cause the rescue of A. viscosus. So here you have none of these other species. Here in each experiment you have one of these other species, two of these other species, three of them, and in this case, all four of these other species are present. So now you look at the abundances of A. viscosus and all of the other ones in each of these experiments. And this is what they found. And again, these are standard kind of plots that you see in this kind of community. I'm going to draw your attention to the abundance of A. viscosus, which is colored this blue here. So notice that in all of the experiments, barring this one, which has all of the other species present, A. viscosus is not rescued. It means, from this experiment at least, that all four of these species are required for this rescue to occur. Right? And how do we know that, that these species are not combined with these species to cause that rescue? Because these LKZ and S. mitis were also there. So they did this other control experiment where they eliminated LKZ and S. mitis one by one and saw that even when both of these were not there, A. viscosus was rescued, which meant that these four species together somehow worked to overcome the negative effect of LKZ and S. mitis to rescue A. viscosus. So what's the cartoon that we are left with? LKZ, strong negative effect, S. mitis, strong negative effect. This species also had negative effect in the pairwise experiments. The other ones didn't have much of an effect. And then these four species together had this strong positive effect, which gave rise to the species structure, the community structures that were then realized. So now this tells you that these high order interactions are present. In this case, it's a fifth order interaction. So if you have to now think about predicting community structures when you contend with these high order interactions, then experimentally this becomes an intractable problem very quickly as the size of the communities rises. So for example, let me show you what it takes to experimentally do what is called as a bottom-up approach, or take a bottom-up approach to identifying these interactions. So you first, of course, have to have, again, these are the experiments that I talked about. If you take a 4-species community, for example, you would need to do experiments where you pick up one species at a time. These are the monocultures. Then pairs. When you do pairs, you get binary interactions. Then you do triplets. When you do triplets, you get ternary interactions. And then you have to do the all-species community. So if you have now n species in your culture, then you would need to do 2 to the power n minus 1 experiments. So this is an exponentially increasing proposition. If you have 10 species, you're doing 1,000 experiments, and so on. So very quickly, this becomes intractable. So this was the question that we were bothered by. So how do you get around this problem, if at all? So we came up with this algorithm, which we called EPICS, which helped us think about this problem. And this algorithm says, we are not going to do all of this business of going bottom up. We'll take a top-down approach where we'll say we predict or estimate what are these what we call these effective pairwise interactions, and use them to predict community structures. So what are these effective pairwise interactions? So in this case, we said we take the monoculture experiments, of course, and then simply take recourse to these leave one out experiments. So one species is left out in each of these experiments. And with just this and these experiments, this algorithm will help us make predictions of this community structure. And in this case, what happens is all you need is 2n experiments. And I'll tell you how this really works. This is our generalized Lotka-Volterra equation. We're going to simplify this by saying that all these higher order interaction terms, we're going to lump into an effective pairwise interaction coefficients. So I'm going to use A primes here instead of A's, and eventually I'll throw the primes away. But for now, just for us to understand it, this is your binary interaction term, the ternary interaction tensor, the quaternary tensor, and so on, and these are going to be replaced by this. So now the problem boils down to finding the interaction coefficients here. What do we mean by these effective interaction coefficients? These tell us the effect that species J has on species I in the presence of all of the other species in the milieu at their steady state abundances in the all species culture. So if we knew that, then we'd be able to predict these culture abundances correctly. How do we find these interaction coefficients? So our thought was to look at the number of coefficients that are involved. Now these diagonal elements, they simply tell us the carrying capacities of the monocultures. So you do the monoculture experiments and you know all of them. And what you're left with is n into n minus 1 of these coefficients, which are the off-diagonal entries. Now if I think about the leave one out experiments, if I have n species in my community, there are n leave one out experiments. Each experiment you leave one of the species out. And then you're left with n minus 1 species in those experiments whose abundances you measure. So you have n minus 1 data points in each leave one out experiment. So the total number of data points we have are exactly n into n minus 1, which is the number of these off-diagonal coefficients that I must estimate. So I have just as many equations as there are unknowns. I solve this problem, I get these interaction coefficients, and then go back to the Lotka-Volterra model here and predict the abundances of the full culture. So does this work? This is the algorithm. Right? So you have these true interactions, you do these experiments, monocultures and these leave one out experiments, and then you extract effective pairwise coefficients and use that to predict community structures. So first I'll show you very quickly how this works in an in silico setting. Right? So I'm going to do artificial experiments. I'm going to pull out these interaction coefficients, drawing them randomly from some normal distribution, and without loss of generality I can set the diagonal entries to -1. Similarly, I'm going to pull out these binary interaction coefficients, pairwise interaction coefficients from another normal distribution whose strength is beta, and then a ternary interaction coefficient, a quaternary one, and so on. So I can keep doing this. And then now, because I know all of the interaction coefficients, I can solve the full Lotka-Volterra system and predict the community abundances, structures, both for the monocultures, for the leave one out experiments, as well as the full community structures. That data is here. So I have the individual monoculture abundances, and then the abundances that the Lotka-Volterra model would have predicted in the leave one out experiments. I can now use this as input to EPICS, estimate these effective pairwise coefficients, and use these effective pairwise coefficients to predict the community structures. So this now, the blue ones are the community structures predicted by the full Lotka-Volterra model using all of the interaction coefficients, and the red ones are what our model would predict using EPICS. And you see that there's a pretty good agreement between the two. In fact, we can now, because we have this going on in silico, we can replicate this many, many times, drawing all these interaction coefficients randomly, and you see that there's quite a good agreement between what EPICS predicts and what the bottom-up approaches would predict. And this inset simply shows you that if you ignore the high order interactions, you'll be quite a bit off from the true predictions. And what is interesting is that the accuracy of EPICS increases as the number of species goes up. Right? So this is now telling you how many species there are and what the error is in the predictions between EPICS and the true Lotka-Volterra system, right? And so you see that the errors are going down. And this happens because as the number of species grows, if you leave one of the species out, the net interactions that the other species are experiencing changes far less than if you had fewer species present, right? And therefore you can see this accuracy going up. So you have a system now which allows you to perform far fewer experiments, number of experiments increasing only linearly with the system size, and gain accuracy. Right? So at least you have a way of making progress where otherwise these bottom-up approaches would be stuck. So we applied this to two settings. One is a published source of gut microbiomes in some of these flies. So this experiment was a really lovely one. In this case, they had these inoculates of bacterial combinations of different, there were 5 of these species that they were looking at, species 1, 2, 3, and they're color coded. And then they did the full factorial set of experiments, 2 species, 3 species, and so on. So they grew these, they had these test tubes where they put these microbes, and then they fed these microbes to the flies, right, which was sterile otherwise. And then they looked at the gut microbiome of these flies. to find the compositions of these species. This is the experiment. And this is their data. So they have monocultures, binaries, etc., etc. So from this, we can, and the full the 5 species community as well. So from this data, we can pull out the monoculture and the leave-one-out data sets, apply epics, and check what happens when we compare it with this data set. And this is our comparison. This is the data set that we took from that experiment. These are the effective interaction coefficients that we predicted. And then here we have the comparison between the experimental data and our model prediction. Looks pretty good. The next, we applied this to this 8-member oral microbial community that I was talking about, which our colleagues at Unilever actually put together. So now, same sort of experiments. You have all the leave-one-out data sets as well as the monoculture abundances. You can feed this to epics, and they also did the 8 species community. So you feed this to epics, you get all these effective pairwise interaction coefficients, and now you can solve for the actual community structures, right? And the blue bars here are the experimental observations, and these orange bars are predictions from this algorithm. So it tells us that we can do a pretty good job, at least at this level, of capturing community structures using these effective pairwise interactions. So now I'm going to switch gears and talk a bit about this other algorithm that I was mentioning to you at the start, which is something that we call quartet, which is to disentangle cooperation and competition in the interactions between species pairs. So I'm going to come back now to pairs, because while we have looked at all of the high order interactions, it turns out that we are thinking about what we call net interactions, and these interactions in turn are nuanced, and unless we understand those nuances carefully, some of these predictions are bound also to be inaccurate. What's the idea here? The net interactions, as also Karthik mentioned, and this is the paradigm in the field. You think about 2 species that are interacting. They may influence each other positively, or they may have one influencing the other positively and the other negatively, and you think about these as mutualism, parasitism, amensalism, and so on. But really, if metabolic interactions are going on, what are giving rise to these interactions? And you can easily tell that there are 2 dominant parts to these interactions. One is that there is competition for resources. So for example, if there's a resource, it gets split into some fraction that goes into species 1, the remaining that goes into species 2, and then they secrete metabolites, right? And these give rise to perhaps positive interactions, perhaps negative also. But if I think about resource competition as being the dominant negative interaction and sharing of metabolites as being the dominant positive interaction, then it's the net of these 2 interactions that manifest as these overall interactions. Right? So really, this cartoon should be something like this. There must be a positive component, and then a negative component, and then of one's influence on 2, and the same thing of 2's influence on 1. But at the moment, there are no methods that can tease these components apart. So our question was, can we do something like this? Can we tease it apart? So we came up with an algorithm that helps us think about this at least, or get estimates of these components. And the algorithm is the following. It is based on a theoretical resource partitioning that allocates all of the resources that are necessary to one of the species for it to grow at the rates at which it is growing in the community. And then look at what remains and use that to describe what happens to the other species in the community. So I'll describe this to you briefly. So we start with what we call growth resource curves. Now this is input data that is necessary, where you take individual species, measure their growth rates as a function of the supply of some resource R, which is the limiting resource. You get something like this. You do the same thing for species 2. Let me call this mu1 and mu2. So you now have these growth resource curves. Let's look at the amount of resource that is supplied to the community. Let's call it RC. And that's true for both these species. At this community's resource rate, if the species were present by itself, this would be its growth rate, and the same thing for this second species. Now, these species, when they're together, they grow at certain rates. Right? It's the difference between these rates that you describe as the net effect of one on the other. This is what is standard, that's measured all the time. So now we are saying, let's look at what resource is necessary for species 2 to grow at its community rate if it were present by itself, and let's call that R2C. This amount of resource is now left over. That resource, let's say, is used by species 1. If it were used by species 1, then species 1 would have grown by itself at this particular growth rate, but it actually grows at this growth rate. So the difference between these two must be the positive effect that species 2 has on species 1. That's the metabolite that's being produced by species 2. So this gives us a measure of the positive interaction. And of course, the difference will give you the negative component. And you can do the same thing the other way around. This is the growth rate of species 1 at the community rate. This is the resource that is required for it if it were to grow there by itself. This is the amount that's left over. That you can use to predict what will happen to species 2 if it were by itself. But then the actual growth rate is this. So this gives you the positive component, and the difference gives you the negative component. So we can use this now. What it gives you is a quartet of interactions, and that's the origin of this name. Let's demonstrate this. Again, so we took this in silico community, and now because we don't have all the growth rates measured and so on, we're going to take recourse to some of these genome scale metabolic models. And it turns out that the results are robust to the specific model that we have used. Of course, we've got to use the more sophisticated techniques that Karthik has developed. But if we do this, then this is what we find. Let's say species 1 and species 2. There is, of course, the net interaction, but then you can recognize that this net interaction is the net effect of a negative component and a positive component. Right? And this is for 2 influencing 1 as well as 1 influencing 2. So if I now take these 8 species, I can produce these quartets for all of the pairs. And what is interesting is the following. Of course, you can now quantify these things. And what is interesting is the following. You can find species pairs that have the same net interactions, but arising from very different components, very similar net interactions. So here you have high negative and high positive contributions, which cancel out to give you this net effect. Here, those contributions are low, but the net effect is the same. So now you'll realize that the response of these communities to environmental changes is likely to be very different from each other. But if you took recourse to net interactions alone, you wouldn't be able to tell that difference. This was the reason for us to recognize that current theories have these limitations, and our predictions of how communities would behave in response to stresses or environmental changes might be inaccurate. So lastly, I'll tell you that these components also give us insights into community behavior. You can now extract these components at all resource rates and ask how community stability changes. So in this particular case, we have done this for these 2 species. These are, of course, the net interactions that you can predict. And you'll see that you go from an exploitation regime to a competition regime. And you can now tell that the origins of these transitions are in the relative changes of the underlying components and not drastic changes that you otherwise envision as happening when you change resources by subtle amounts. Right? So you get insights into community behavior. I think I'm running out of time, so I'm going to stop here by telling you that, in summary, high-order interactions may exist in multi-species communities. We have evidence of that from some of the experiments that we have done. EPICS subsumes these high-order interactions into effective pairwise interactions and helps us predict community structures. It's efficient, it reduces experimental effort, and it's accurate and scalable to large communities. It, of course, has limitations. It assumes that there is a single stable steady state, and this is something that we are still thinking about. It also assumes that species abundances are all non-zero. Of course, we have now overcome this. I urge you to go to the poster that Adil, one of our group members, has, where he has developed an alternative algorithm. It's actually a generalization of EPICS, which uses Moore-Penrose pseudo-inverse to overcome this problem of species going extinct and still applies EPICS to get accurate results. And then there's this algorithm called Quartet, which helps you disentangle positive and negative components of the interactions, and it has its abilities to offer insights into community behavior. Thank you very much.
Thanks, Prof. Narendra. So we do have time for questions.
Narendra, great talk, great work. I think I'm going to repeat Revati's question here, right? So is it possible for you to infer in some way, and using the data at hand, can you set aside species and look at the effect of unknown species in some fashion?
Unknown species?
The unknown players in the mix, right? So players who exist, but we don't know that they exist, but what is their contribution to the community community dynamics?
So we haven't thought about that. At the moment, we have been thinking about communities whose species are known to us, and we are trying to manipulate them to produce synthetic communities and so on. One can take these approaches where we think about effective background.
Yeah.
One can think about those. There's probably some whole bunch of species which has an effective interaction that
because you have already subsumed some interactions into effective interactions, so can this somehow be extended?
So we could create another fictitious species which involves all of the species that are not known and think about its interaction with all of the existing species and ask if one can draw inferences about what these background interactions might be. Interesting question, we have not done that.
Yes, Niranjan.
Hi.
Niranjan, hi.
Very interesting talk.
Thank you.
Good to finally see it fully fleshed out. So I have a question. So for Quartet, right, what kind of experimental data, from what you described, it sounded like you're just taking genome scale models and combine doing the individual and the co-growth condition tests. But if I could generate experimental data, what kind of experimental data would inform these models?
Right. So the genome scale modeling was only a way to substitute for the experimental data that we currently don't have. The data that you need are these growth resource curves for the individual species and then the growth rates in the communities.
What is growth resource? Which resource?
So,
There's so many resources.
No,
right?
for example, you take whatever medium you have. For example, let's say people say glucose as, let's say, the limiting resource or some predominant resource or carbon source that a particular community uses. Then if you can vary the amount of that particular entity that is responsible or dictating the growth rates of the species, then you have this way of doing those experiments where you systematically increase or decrease the amounts of that limiting resource. And then it has to be a limiting shared resource. That's the source of the competition between the species. If it is not shared, then this theory doesn't even need to be applied.
But if there are multiple limiting shared resources, you would want to measure each one of those.
Right. So you're right. Multiple limiting shared resources are something that we haven't accounted for. Normally limiting, when you say limiting, it's usually one. But if there are 3 or 4 that are simultaneously necessary and they can all be simultaneously limiting, then that's an added complexity that we have not accounted for in these theories. Yes.
Excuse me, here. Yeah, it's a good talk. And I continue with Karthik's question about unknowns. So here you are validating with the cultures because you are putting what cultures you know how much it is you are putting, and then you're counting, CFU based analysis, you validate. How do you validate the unknowns? Because we do not know what they produce. We don't know more than 50% of the genome if you know all hypothetical protein, and how do we do that?
So, great question. My answer to that is even for the known ones, we don't know what is going on. So at least we are making some progress there. Okay. And to take it to the unknown ones, I think there's another leap that one has to make, perhaps through the arguments that Karthik is suggesting we take.
Okay. The validation is the biggest issue,
Indeed. So at least here we have validated it for the known ones, right?
And still, are you able to reproduce it again and again? Yeah. Because these seven species, because biology is, even with a small change in pH and then other things, you cannot get the same thing.
Sorry, it's taking me a while to go back, but there are error bars on everything there. So they're all repeats. I don't know how to go back.
I agree with that.
Okay.
So it may be Dikshit lab have a very good hands, but
not even my lab, it's Unilever. They have very good hands, by the way. Yes.
Okay.
Yeah.
It's not robotically manipulated, right?
No, you can do robotics, but I don't think there is any. So there are uncertainties which are significant, right? There's at least sometimes even a 2 orders of magnitude variation in the abundances of individual species that you can measure across repeats. But we factor that in the model because we use data that also has similar uncertainties and then allow uncertainties to play out in our predictions also.
This is monoculture, this one, right?
No. These are the full species, full community abundances, yeah.
Thanks. So I'm just curious with a follow-up question to what Niranjan asked. So since you used the genome scale models in your Quartet, which also gives you the resource breakdown internally within the species, right? So have you had a chance to look at why there is a increased cancelling out effort, although the net interactions is the same? How that is different?
No, that was a peculiar example in the sense that you have these two species pairs which have these different positive and negative components whose resultant turns out to be the same. Mm-hmm. It's not always the case. It just happens to be the case.
I see. Okay. Any more questions? Yes. Sorry.
I was wondering if you'd be able to predict what types of mutations in which of the pairs of species would be most effective at changing interaction dynamics a lot in the second part, the Quartet part.
It's a great question. I think we don't know the answer to that question, mainly because you have to figure out what those mutations affect by way of metabolism, because these are all metabolically underpinned predictions.
Yeah.
I think one has to go back to models like what Karthik and all have developed to think about what genome scale effects individual mutations have. And I'm not sure that is very well understood unless you have a fully curated genome scale model. Or else you've got to think about some very specific reaction or pathway that is influenced by a particular mutation and ask whether that is influencing your predictions in a certain well-defined manner.
No, I was thinking more broadly. So for example, you have these net effect is positive, let's say, but you can have the underlying positive and negative effects be very large or small.
Yes.
So I'm wondering whether a change of, a certain change in the growth rate of a certain species in the presence of the other and so on, if there's something non-intuitive, maybe I'm not being very clear, but anyway, yeah, what you said about the mutations themselves makes sense, yes.
Right. No, I have to think about that. I haven't thought really about how to map one, like what he's asking also, right? How do you take one species pair which has large components and convert them into something that has smaller components but still keep the net interactions the same?
Essentially, would that redefine how we think about, say, parasitism and mutualism and so on, right? I mean, or should we use quartet to classify these things slightly differently?
In fact, that's the theory, that's the thesis that we have, that taking recourse to just the net interactions may be too simplistic. Yeah.
Sure. In the interest of time, so let's thank Narendra once again.
Thank you very much.
So there are also some questions from anonymous people you may think of.
I may think about them. Thank you.
Thank you.
We'll now have the flash talks. I'd request the participants to be seated nearer to the stage. First talk is by Dr. Revathi.
Good evening, everyone. I'm Revathi Krishnamurthy, currently a research scientist at Evelyn's Group in Quadram. And I would like to thank organizers for having me here. And this particular work is very close to my heart because I'm a mom and a phage biologist, and getting asked by my children, Mom, did I inherit my phage from you? And something like this is very interesting to discuss with my children, at least for that. And then there are a lot of studies that, far and few studies have actually talked about maternal influence in shaping up the gut microbiome of babies, and with contradicting results. Some say, yes, there is maternal influence in shaping up the infant virome, and some do say, no, it is not. Definitely in the later on stages in the baby's development, we do see that the environmental factors come into play, but what is happening during that early lifetime is something I'm going to focus today with you all. So for this, answering this, we have a longitudinal study going on in Quadram called PEARL, Pregnancy and Early Life Study, where we have samples from baby and mother, right from when the mother is from her first trimester till the baby is 24 months old. And this is just the breakdown of the subset of
samples that we used from the study, roughly about 15 baby mother pairs with total 137 stool samples. And this is the workflow that Evelyn and Karthik have spoke about yesterday, and I'll be very happy to talk to you about if you want to. Briefly, after extraction of the total nucleic acid, after enriching the viral particles from the stool sample, and after extracting the nucleic acid, total nucleic acid, which I mean both RNA and DNA, and we did the sequencing and did the basic QC, got the assembly done, and used multiple viral mining tools that involves both K-mer-based as well as homology-based algorithms to mine the viral sequences from those assembly, and then further clustered them using, based on the average nucleotide identity, and further put the check-V cutoff, like most of you know, or yesterday we heard from Evelyn and Karthik. And we used only high-quality max for our analysis. And on top of it, actually, we also had put the abundance threshold to delineate the signal from the noise. And with this, we had our VOTU table, which showed majority of the VOTUs, what we have in our data set is double-stranded DNA. So in this talk, I'm going to address what is the influence of maternal virome over the babies. And the first thing I asked is there, first of all, a stable virome in the babies from infancy to the toddler stage? The answer is no, because during the early time point when the baby is developing closely with the mother, we see a lot of drift in the number of VOTUs, and it is highly abundant at the month 4 compared to the rest of the months, and pretty much the adult virome, mother's virome is stable here. And I do see that this month 4 virome is pretty much very different compared to the rest of the months in the babies. And definitely, infant's virome dynamically changes over that early time point. And the main point I want to convey here is that majority of the early virome is a shared component between baby and the mother pair across the realms, actually. So I would like to draw your attention towards this dotted portion of the bar, which shows the shared component, the VOTU that are present both in the baby and the mother. And we see the shared components we see across the realms in the virome. And what you also see, what I want to say here is basically during this early time point, week 3 and month 4, majority of what the baby has is coming from the mother, and what you see a little of this unique VOTUs that the baby has, that is also very low abundant one, if you remember yesterday's slides from Evelyn. So what I want to say is, like, why is that the case? How this virome shaping up is regulated? Definitely looks like there is some sort of regulation going on, and what exactly is the importance of such regulation and things like that would be very interesting to further probe upon. And I also see that this is an example of one baby mother pair shared component, and what I see is some of the VOTUs are quite persistent. The VOTUs that I see in the trimester 2 in the mother, I could see it till the baby is at least roughly 2 years old, and some are just co-occurring. Just because we had, for this particular sample, month 4 samples was sampled for both mother and the baby. So some are just co-occurring and some are highly persistent. It'll be very interesting to see what these persistent VOTUs are and what are they. And moreover, I leave this slide for you to have the summary, but what I also see is some of the VOTUs, roughly 2% of my data set is present and highly prevalent across the Norfolk population. At least 9 out of 15 pairs what we have has those VOTUs, and some of the VOTUs are reported in some other independent metagenome data sets as well, which basically it's called a UHGV database, which cataloged all the early gut virome VOTUs, and we do see such VOTUs here, and it'll be very interesting to look at them as well. What are they and what do they do? And further, actually, I would love to use the metagenomic data set that our collaborator has, and to see more of the prophage content of the same sample and see what exactly is getting changed in this equation. And I would love to investigate the role of this shared VOTUs in shaping the human gut virome. Thank you for the opportunity, and I would love to thank my whole group, and especially Ryan, who helped me or who worked with me together in giving you this story, and also definitely my children for letting me fly. Thank you all, and I would like to take questions.
We can have just one question, if there is any. No question? Oh.
From the previous talks, I understand that this crass phage is one of the most abundant phage that we have in the gut virome. Can you give a small context on when this crass phage is basically coming up in the baby?
That's a good question, actually. But with this, what I currently have, it should be something that starts the later stages of the development, and I'm yet to dig and if I see anything of that crass phages or not. So it's still in the preliminary stages. So if I see, I'll get back to you.
Thank you, Dr. Revati. Next is Pavan.
So hi all, myself Pawan Kumar, PhD scholar from IIT Madras, and I thank the organizers for giving me this opportunity to present my talk titled Sprint Gap Filler, an efficient gap filling algorithm for large-scale metabolic models. So this is a quick outline of what I'm going to talk today. So quick introduction about constraint-based metabolic models, which previous speaker also had an emphasis about, and why we need to gap fill in metabolic networks, what's my problem statement is, and a short overview of sprint gap filler with the results and conclusion. So as we all know, metabolic networks are nothing but set of biochemical reactions and metabolites, which helps us to generate energy, to convert food to building blocks, and to eliminate waste. And one of the widely used representative or mathematical model for this metabolic networks are what are called constraint-based models. And typically, these three are the variables that has been used to represent a constraint-based model, which are stoichiometry matrix, gene-protein reaction rules, and the constraints, which tells, which gives the thermodynamic constraints for a given reaction. And why we need to do gap filling in metabolic networks. So here, gap filling always comes throughout the life cycle of a genome scale model. So here I'm just showing two applications, one with respect to genome scale model and also one with respect to community, microbial community gap filling. So a given genome will always have evidences about what are the reactions present. Now let us say if we have evidence about the presence of A to D reactions, this one or two reaction doesn't make a model. Like we need to see what are those reactions that helps in consumption of D, and also we need to fill the gap such a way that we have to identify what is the reaction that produces A also. And why, first of all, these such gaps occurs because of this misannotations, or there could be some transport or spontaneous reactions which are present. And how do we get them back to our model is where gap filling comes into the picture. So these are some of the important requirements for any gap filling algorithm. One is if I define some predefined reactions, like in the previous slides, these are my predefined reactions, which I would like to call it as a core reactions. Those core reactions has to be present in the final model. And for remaining reactions, if I were able to derive some weights based on sequence similarity or some other approaches, and I should be able to weight the remaining reactions. And media or metabolics, if I have evidences on media or the metabolomics, I should be able to constrain based on that too. And the final model should be minimal. And if you are trying for a large scale model like a community scale model, it should be scalable algorithm, and it should be a consistent metabolic network, saying that whatever reactions present in the model, final model should be able to carry some non-zero flux. And not only one pathway, if there are few alternate pathways that could that is able to satisfy that non-zero flux through the core reactions, algorithm should be able to list out those two. So this is a problem statement in terms of mathematical terms, where S is a stoichiometry matrix, and given reaction set and irreversible and the constraints, which are predefined variables, and the core reaction set, how do we get that output, which is n, and n has to be minimal. So I'm not focusing on this mathematical or algorithmic part, but I will happy to discuss if anyone is interested on this. So in short, there are three different LPs which are core to the sprint gap filler: LP forward, LP reverse, and LP minimal. So what it does is, LP forward push the forward flux through all the set of core reactions, and LP reverse again push the flux through core reactions, but in a reverse direction or a negative direction if it is a reversible. collect the evidences of what are those reactions which had a non-zero flux along with the core reactions. And once we have that, reduce the size of the network. Reduction means either reduce the count or reduce the sum of the flux through the remaining reactions. So this is in short the sprint gap filler algorithm. And the first results I'm showing is with respect to the time it takes to build the algorithm. So FastCore and SwiftCore are previous existing algorithms which are again LP in terms of gap filling. So and that's why we are compared with this in terms of both runtime and number of solved linear programs here. So as we can see, sprint gap filler is the current fastest algorithm to get the minimal network. It is comparable with respect to SwiftCore, but SwiftCore cannot at times handle this media constraints. So that is one limitation of SwiftCore, but sprint gap filler is able to handle them at the same time be the fastest one. The second problem statement I'm mentioning here is on getting the list of block reactions, which is an equivalent problem statement. Like say here, particular reaction A to D is a block because there is no other reaction which consumes D. And here, this is the results just I'm presenting here, where we show again the time or the speed of the computation in terms of runtime and also the number of solved LPs. So here the green color as it shows is the currently we have the fastest algorithm to detect the block reaction in a genome scale model or a constraint based model. So conclusions, we present the two different algorithms, sprint gap filler and sprint CC. One is for gap filling, another one is to get the identify the block reactions. And currently we are focusing on scalability of this particular gap filling algorithm, scalability in terms of both count, where we are trying to build a whole bunch of single cell models, and in terms of size, where we are trying to gap fill one complete microbial community in a go. Finally, I would like to thank the two supervisors for this work, Dr. Neerabhat and Dr. Radhakrishna Mahadevan, and my colleagues who have been part of this work, Subhashri Sridhar and Noor Alsmedi. And thanks IBSE and MITAX for funding this project. Thank you.
Any question? No. Okay, we'll move on. Anna will speak next.
Hi all, and thanks for the brilliant opportunity. So today I'm going to very quickly expand on one of the case studies that Venkat introduced this morning and that I had the pleasure to work on over the past few months. So it's quite a unique case of persistence of bacterial cells, not spores, in NASA clean rooms. Particularly the work started and was prompted by the isolation of Acinetobacter johnsonii in NASA clean rooms and then evolved to a complete characterization of the species across different settings. So far, there is little evidence in the literature of persistence of Acinetobacter johnsonii as a gram-negative non-spore-forming coccobacillus in high-stakes controlled environments, including NASA clean rooms, of its occurrence in opportunistic infections and of carbapenemys-producing strains. And our knowledge, however, of the mechanisms underpinning the increased adaptability of Acinetobacter johnsonii to extreme growing conditions have still a lot of gaps. So the objectives of the study were to address these gaps and to explore Acinetobacter johnsonii's potential as antimicrobial resistance reservoir. To do so, we performed a comprehensive genomic analysis that included 22 isolates of Acinetobacter johnsonii that were recovered from the clean rooms during and after testing and assembly operations of NASA Mars Phoenix lander. We'll address them as PHSF isolates. And one carbapenem-resistant strain, we'll address this as the Galway isolate because it was associated with patient colonization in Ireland. So we received these isolates, we confirmed species typing, and performed antimicrobial susceptibility testing. And then we went through a comparative genomic analysis that included, for broader genomic context, all Acinetobacter johnsonii genomes available on NCBI. And finally, we tracked the persistence of Acinetobacter johnsonii in NASA clean rooms through genome to metagenome mapping. Based on core genome phylogeny, PHSF isolates cluster into a monophyletic clade that diverges from the main species lineage, suggesting the development of genomic features that may be linked to increased tolerance towards extreme growing conditions. Most of all, complete resisome characterization, our analysis allowed to confirm and uncover the absolute ubiquity of beta-lactamase gene, which were found to be intrinsic to all the genomes of Acinetobacter johnsoniae. And this underscores the potential of the species as antimicrobial resistance reservoir, which was further underscored by profiling the Galway strain as multi-drug resistant and particularly carbapenem resistant. The virulum characterization then allowed to uncover 2 complete efflux pumps in PHSF originating isolates that were extremely conserved in these isolates and mostly absent in the other genomes. Metabolic prediction based on the genome assemblies also allowed the identification of a unique racemase function involved in purine metabolism that was extremely conserved in all PHSF isolates and was found nowhere else in Acinetobacter johnsoniae. Again, all these features can be linked to persistence in oligotrophic and extreme growing conditions. Finally, genome to metagenome mapping, specifically mapping back to the metagenomes collected from the clean rooms, allowed the identification of genomic signatures and a minimal population of viable cells of Acinetobacter johnsoniae up to 10 years post-initial isolation. In conclusion, our results suggest that PHSF originating isolates might have deviated from the main species lineage due to the emergence of genomic features that are linked to their survival in extreme growing conditions. Further, we highlight as a whole species the potential of Acinetobacter johnsoniae as perhaps understudied AMR reservoir. And although our hypotheses still need further research to be validated, these findings will help evaluate and manage the contamination of Acinetobacter johnsoniae across clinical, terrestrial, and extraterrestrial environments. Thank you all for listening.
Hi, I have a simple question. So what do you mean by metabolic prediction? Is it identification of metabolic genes?
It's based on functional annotation of the genomes of nucleic acid sequences. And then we use eggnog mapper and KEGG charter to map these functions to databases so we had 200 of these metabolic maps I show. And yeah, that's just based on functional annotation of the genomes.
Can I go? Hello. Hi. Yes, over here.
Okay.
So, did you see it in any other environment, rather like anything else from any human isolate, any other environment, extreme environment, any isolate from extreme environment, and what kind of environment it would be?
There is very little in the literature. So, well, one is the paper published by Venkat and his colleagues about the recurrent isolation of Acinetobacter from the clean rooms, which is the ones we worked on. And then there is isolation of Acinetobacter johnsoniae from the environment, mainly from soil. And there is very few cases of opportunistic infections and carbapenem resistance, but we're talking of 2-3 instances.
A follow-up, so how far this?
Yeah, there is. Yeah, we worked on a total of 112 genomes.
No, so if there is any other environmental isolates, how far they are from like any extreme environment like clean rooms? That is my.
They're all environmental isolates, essentially. All those available on CBI, bar a few.
Okay, thank you. Nice talk.
Yeah, hi. A very quick question. Yeah.
Okay.
Yeah. So in your AST profiling, what kind of MIC breakpoints have you used to define whether it is showing resistance or susceptibility to different antibiotics?
We use standard EUCAST MIC breakpoints, though it's a bit tricky because Acinetobacter johnsoniae doesn't grow at 37, so there is need to establish proper breakpoints for clinical purposes. It's a bit tricky to interpret the results because they don't grow at 37, but yeah, we stick with the guidelines, the UCAS guidelines.
So with Johnsonia and with other extremophiles, we have the problem that the interpretive criteria that UCAST and CLSI are using are not applicable because they don't grow at 37. So yeah, we are in contact with UCAST, given Johnsonia is a clinical pathogen now, to see how we can amend this criteria and maybe suggest for hospitals as well to use 30 as a growing temperature because it doesn't grow at 37.
Thank you, Anna. We have Cody next.
Thank you, everyone, for the invitation to come speak. I am a PhD student in the lab of one of the Karthik's. So I'm going to be telling you about a method that I've developed called Protein Set Transformer, which is a genome language model or deep learning model. It's really to learn more from what genomes encode functionally using purely genomic data. So I think something that we're all kind of generally familiar with is that genome databases, especially today with metagenomic sequencing, are exploding in scale. And so what we can see here is that there is exponential increase in particularly the number of viral genomes in genome databases, especially uncultured viral genomes. And it's not just enough that we have a large number of viral genomes, but they're also extremely genetically diverse. So just to give you a little bit of context, these 16 million genomes encode around 120 million proteins. Of that 120 million proteins, around 60 to 90% of them cannot be assigned function. So there's a large amount of information that we don't actually know about viruses and microbes as a whole. And so really the goal of my work is to build sort of a foundation to be able to learn more generalizably and more sensitively using purely genomic data. And so a little bit more about what I mean by that. If we look on the column on the right side, we have these massive genome databases. We can train a very generalizable foundation model, which I call Protein Set Transformer or PST, to leverage all of the information that is in these large genomic databases. And then in the future, we can apply these foundation models to more specific tasks that people are probably more interested in, such as protein annotation or host prediction. This is similar in what has been done for foundation models like GPT, which have been trained on large text databases. And perhaps the most famous application I think that we're all aware of for this GPT foundation model is ChatGPT, which I think needs no explanation on the utility of what ChatGPT can do when extended from the GPT foundation model. So PST, like I said, is a genome language model. And just to give you a little bit of a sense about language modeling as a whole, language modeling really operates on this idea that there are words that compose sentences, and the meaning of the sentence can be informed not just as the sum total of the words that are written in the sentence, but also there are these interactions among the words in the sentence that can give rise to these sort of emergent properties of what a sentence can mean. And so that's kind of very abstract, thinking more a little bit in biological terms. So I kind of model genomes as sentences composed of protein words, but proteins can also kind of be thought of in the same way. So the way PST works is to think about both proteins and genomes as sentences. And so kind of what that looks like is if we take all of the viral proteins, all of the proteins from a viral genome, we can individually use protein language models for each protein to get a sense of the protein function and structure as a function of these combinations of amino acids within each protein sequence. And that gives us really rich information about proteins. And then subsequently, we can use this at a genome scale to learn what does genome context allow us to better understand about the proteins, and subsequently, what can we infer about the roles of the genomes as a whole. And so to train PST, I wanted this to be very generalizable and useful for future applications. And so I trained PST with a triplet loss objective, which can be seen here, where the basic idea is that we want to optimize the closeness between genomes that are more related to each other, and we want to repel genomes that are unrelated to each other. And so I want to emphasize that the model is not predicting any type of label about the virus. It just wants to learn these types of relationships. And when training the model to learn these types of relationships, if we look at intrinsic measures of genome-genome relatedness, for example, average amino acid identity or shared protein content, we can see that if we cluster genomes based on PST representations, that on average, these genome clusters have higher average shared protein content compared to the input, as well as even if we just clustered genomes on the basis of AAI in the first place. So this is great to see that PST seems like it can understand these genome-genome relationships, but this understanding of genome-genome relationships is also built upon an understanding of protein function and organization. And so just to give you a quick idea of what that looks like, so what we're looking at here is each protein is a single dot in these plots. The colors indicate the protein cluster, and the shapes indicate the protein functional category. And so what we're comparing is on the left without genome context and on the right with genome context, so PST. And if you look at a lot of these plots, and you can also summarize this quantitatively as well, we start to see that there are functional modules that get detected when we consider genome context, so structural modules, late gene modules, and so on. And there are also annotation improvements. So you can see these small circles that are hypothetical proteins that are clustering often with proteins of known function. And so just to give you some other brief highlights, especially thinking more about these hypothetical proteins, so PST can also detect protein structure without directly computing the protein structure. And this is really significant, especially again for these proteins that are not annotatable with current methods. And in addition, thinking about this idea of using these foundation models and then applying them towards specific tasks, PST can also be applied towards host prediction, and that seems to work very well. Especially even compared to pre-existing host prediction methods. And so finally, I kind of want to just come back to this idea of foundation models and think about, I've showed you very briefly some applications of PST and some things that it's learned. In PST and genome language models as a whole, they seem to be very useful and can be applied, I believe, to a variety of different tasks that are common to genomics applications and genomics studies. And if we can do better to really get at, some of these things like protein annotation, that really is going to extend what types of new biological insights we can gain about our microbiomes and the roles that viruses have that we've previously have not been able to really fully elucidate. And so with that, I'd like to thank, again, the organizers and the lab, and happy to take questions.
So very good talk. So how do you predict the host? Is there any, you said in one of the block there, host prediction.
Yes. So this particular one, this is just a proof of concept study. Do you mean just in general, or?
That is the one I'm asking.
Yeah.
The host prediction that you have, how do you do that?
So the framework that I use is to model host prediction with ecological graphs. And so one aspect that PST really helps with is if we can understand these viral-viral relationships better, that can help inform, for example, if you knew the host already of one of these viruses, and then now you can more sensitively detect what a more related virus is, that can give you some insight into what the host might be.
I'll talk to you later more.
Okay.
Sorry, I had a quick question about especially RNA viruses, which are known to have important secondary structure features which affect their function in many, very strong ways. So can you imagine how you would extend this model to incorporate that kind of information?
Yeah, that's a great question. I get asked this question often, especially for RNA viruses and eukaryotic viruses, where it's not trivial to computationally predict where the open reading frames are. That's, I would say, I guess, one of the major limitations of this work, because it's just challenging to do that. So if we don't have accurate open reading frames and so on, then we can't really model it in the first place. So that's kind of all I can say.
Okay, thank you, Cody. We have Adil next.
Hi everyone, my name is Adil. I am working in Professor Narendra's lab in IISc Bangalore. So today I'll be talking about my research work, predicting microbial community structures using effective pairwise interactions. First, I'll give you a quick background. So designing synthetic microbial communities has important applications in healthcare and biotechnology. So it is important to predict the community structures in order to design synthetic microbial communities. So by structure, I mean the different species present and their abundances. To predict these structures, we need to understand the interactions between the species. So there may be pairwise interactions, ternary interactions, quaternary interactions, and so on. So the most commonly used approach that is used to predict infer the interactions is the bottom-up approach, where the number of experiments that needs to be performed is around 2 to the power n-1, and this increases exponentially with the increase in species size. So our group has come up with a top-down approach, which Professor Narendra has previously mentioned, called EPICS, where these higher-order interactions are subsumed into effective pairwise interactions, and we need data from two different sets of experiments, leave-and-out cultures and monocultures. Here, the number of species combinations that is required to be analyzed is just 2n, and the species combinations increase linearly with n. Now, I'll quickly go through the formulation of EPICS. So here, you see the time evolution of the population is described in this form, Ni into Fi of n, where Ni is the population of species I, and Fi of n is the per capita growth rate of species I. So using generalized Lotka-Volterra model, this resembles the bottom-up approach, whereas EPICS subsumes all the higher-order interaction terms, Bijk and Cijk and so on, into effective pairwise interaction term, Aij effective. Now, we use the leave-and-out data, leave-and-out equations and monoculture equations to calculate these effective pairwise interactions and hence the full species abundances. Now, one of the major limitations of EPICS is that we see that there are species extinctions in the leave-and-out subcommunities. So EPICS algorithm cannot handle because when a species Ni becomes extinct at steady state, Ni is 0, however, the per capita growth rate, Fi of n, is equal to 0, no longer holds. So, if m such species are missing, then we have n into n-1 minus m equations. However, we'll have n into n-1 variables because effective pairwise interactions always exist. So, we modified our algorithm and call it generalized EPICS or GEPICS, which solves a set of underdetermined system of equations using Moore-Penrose pseudo-inverse. Now, we tested our algorithm using in silico simulations. We created scenarios where in the leave-and-out abundances, we have species extinctions, and we applied GEPICS and obtained the effective pairwise interactions and hence the abundances, and the predicted abundances agree well with the conventional bottom-up estimates. We repeated this procedure for 100 realizations, and then we observed that the root mean square of the relative error is approximately 0.3, which is reasonably good. Now, with the increasing number of missing species in the leave-and-outs, however, the error increases, but with increase in the species pool size, this error is drastically reduced. Now, we validated our model with two sets of data from literature. One is an 8-species soil microbial community, and the other is a 12-species synthetic human gut microbiome, where there are species extinctions in the leave-and-out abundances. And we applied GEPICS and observed that the predicted abundances using GEPICS matches well with the experimental values. And we also accounted for the uncertainties in the experiment, as you can see in the fourth plot in the bottom. So in conclusion, GEPICS overcomes a major limitation of EPICS and thereby enhancing its scope and applicability. Thank you. I would like to thank my professor Narendra Dikshit for giving me the opportunity to pursue this project, and I would like to thank IBSE for giving me the opportunity to give this flash talk. Thank you.
Questions? No. Thank you, Adil. So we are done for the sessions for today. For all the poster participants who haven't got their posters evaluated, please be at your posters for tomorrow's session at 11:30.